us to introduce the programs which are having more with skill component than the theoretical component. So in this juncture, in this context, definitely all these days we have been going with all conventional ideology, that is chemistry, botany, physics and zoology. These are the four basic sciences for the nature. To study the nature, these are the four basic sciences. So now the NEP as well the policies of the government are asking us to promote the applied subjects of these particular things. So in this context, definitely chemistry has a wide scope to merge with any subject. Like chemistry can be with the physics, chemistry can be with the biology, chemistry can be with computer science. Like that, lots of scope is there for the subject chemistry, because without chemistry, nothing is there. Chemistry is everywhere. Chemistry is within us. If the reactions are stopped, there is no life at all. That is the reason. Definitely, the chemistry is one subject that can be do anything with any other subject to attract towards the subject of chemistry by introducing multidisciplinary programs in our colleges. So I definitely congratulating here. It is my responsibility to congratulate, because the Department of Chemistry is taking so much of pain and strain to organize this particular program. Definitely this is going to be used by any college or any institution to introduce the new programs in coming academic years. Definitely our government of Telangana, in our state, there are nearly 18 to 19 autonomous colleges are there. These autonomous colleges have got their freedom to introduce the new programs. So definitely whatever the conclusions are definitely useful to the students for their bright career. So I once again congratulating the head department of chemistry, Yadaya Garu, and the vice principal Shukla Garu, and Dayanand, Vasanta, and Busi Anil, all the faculty members of chemistry, as well the members of City College who are totally involved in this particular program to do this in a grand success way. So I wish all the participants and the students enjoy these 2 days in the City College and make it a grand success. Thank you, one and all.
Thank you, sir. I deem it a great privilege to welcome Dr. C.S. Subramaniam, the guest of honor, Professor of Chemistry at IIT Hyderabad. He was awarded with the Research Excellence Award in 2021 and Fellow of Royal Society of Chemistry 2020. Now I request him to deliver his speech.
Respected dignitaries and guests, my friend Srinivas Reddy, Mr. Bhavavaskar, and other critics who are sitting on the dais, and participants of the dais. At the outset, I would like to congratulate this college for organizing such an event. The main reason for congratulating is twofold. First and foremost, I think this college happened to be one of the celebrated colleges. Maybe, if I'm correct, probably this has 100+ year old history. So, and at this juncture now, the theme of the conference, what has been chosen, is really apt. Basically, now it is trying to bring all the interdisciplinary areas onto the platform. I'm sure that all of you are aware at this point of time, everyone is in the entire world, we are worried for 2 molecules. One is how to tackle carbon dioxide, another one how to get green hydrogen. So if you look at these 2 molecules, I think CO2 can be a platform chemical, because if you look at even the petroleum refining or even the crude oil, the basis of it comes only from the carbon dioxide. So the way nature has a peculiar way or a straightforward way of fixing the carbon dioxide, I'm sure that at the end of this conference, I've seen that there are some talks which are basically concentrated on green technologies, carbon dioxide handling, this kind of things. I'm sure such type of conferences are really needed, basically to bring many people onto the same platform. There are eminent people who are going to talk, okay, so who are going to impress all of you that what can be done in these areas. I mean, when somebody talks about sustainability, I think it's very easy to talk, but very difficult to follow. I think some textbook gives a standard definition of what is sustainability, how to follow, what are the principles of green chemistry, what is circular economy, all these things. But at the end of the story, if you look at as a consumer, or if I look at myself as a consumable, if somebody says that, yeah, you need to put an additional energy basically to our additional investment of energy to get some deliverable, I'll take a back step, because it's much easier to talk from this audience or from this platform that, yeah, something can be done and something has to be done. But I think unless such type of conferences impress you on the latest developments and the possible directions in which the research can carry on, I think one should take the inspiration from these things, especially the budding students who are sitting here. Probably you're all the next generation, or you're also going to be the research scholars, or you're going to take the research forward. So I request the college and maybe to impress the state government so that you can quickly start your master's program, because masters in chemistry definitely nowadays I think there are, so though it talks about the horizons, especially like if you really want to collaborate with someone, I think it's the next door for you, like, okay, because science is so developed and you need to just bridge the connection between two different disciplines. Thanks to the latest developments that are happening, either in terms of the nano or in terms of the information or the biotechnology or the artificial intelligence which we are talking about, I'm sure this is not too far that all of us are going to celebrate the success in the near future. And I thank the organizers, and actually I've gone through the brochure, I glanced through the brochure. I think a lot of participants and a lot of enthusiastic audience are going to be here. So I request all of you to please pay attention, especially to the students who are sitting here. If you have any doubt, don't hesitate to ask. I think that's the only way to go forward. I think so, don't hesitate to ask anything. I think probably this is the right platform, I'm sure about it, and I'm also sure that the deliberations are going to be really useful to all of you. So with this, I thank the college for inviting me, also for organizing such an event, and many, many congratulations to all the organizers for this. Thank you.
Thank you, sir. Now, I consider it a great honor to welcome the special guest, Manibhushan Kotala, PhD, Assistant Vice President at Laurus Synthesis. His group has successfully developed and commercialized several NCF molecules for pharma and biotech sponsors. Dr. Mani has greater than 18 years of experience and authorized multiple patents and publications. He obtained his PhD in organic chemistry from IICT Hyderabad, and he has worked at Johns Hopkins University and the University of Kansas in US. For his postdoctoral research, I request him to kindly deliver his valuable words.
Good morning, everyone. Respected principal, my friend, Director IIHCS, and other dignitaries on the dais, Professor from IIT, Subramanian, and delegates participated from various colleges, industries, academies, and students of City College. Thank you everyone for inviting me to today's seminar. And Professor Dayanand was behind me to participate here in spite of our busy schedules to make interactions between industry and academy. That's the motto for AMIA. Thank you, Dr. Dayanand, for continuous follow-up with me to participate here. And I am very much delighted here to learn that the City College is the topmost college, fully occupied with all the students without any admissions. The topmost first college in Hyderabad. So I learned this today, and I am very much delighted to be here in a top one college of Hyderabad, government degree college. And also, the college is well known for 100 years of education for various students across the Telangana, and very good, talented students evolved from this college. To example, one of my colleagues who works with me, Ravinder, is here. He is hailed from this college from degree. So I consider Ravi as one of the best colleague with good knowledge in chemistry. Whenever a tough project comes, I give that project to Ravi. Thank you, City College, for providing such a intelligent and knowledgeable students. The chemistry, Hyderabad is famous for mainly three things. I think most of the people know. The first one is biryani, everybody agrees, right? The second one is IT. The third one is pharmaceutical industry. Many people doesn't know that last 10 years ago, pharmaceutical industry revenue was topmost from Hyderabad, export revenue. When compared to IT industry, now IT industry has overtaken. Now it is our turn as a chemistry students or people who are working in chemistry, we should bring that, charisma to chemistry from Hyderabad and ensure that we all work together to make the revenues from pharmaceutical industry should be number one compared to IT industry. Lot of opportunities are there. Even today, 50% of chemicals we procure from China, we import. So I request all the students from City College, work hard, become entrepreneurs, become good scientists, develop quality chemistry, and try to minimize the purchases from China, develop the chemistry and to manufacture in Hyderabad and India under the leadership of Indian government, like Making India concept and Abhivruddhi Bharat. In that concept, we all need to work hard. We all need to cherish in our field, and we have to produce all the chemicals from India itself, and we should minimize the imports so that the Indian economy will increase and will become number one in economical status across the globe. So that's where all the students and young generation should work and cherish yourself in the field of chemistry. Not only in chemistry, any field one need to cherish. But chemistry opportunities in Hyderabad are very vast because of so many pharmaceutical industries. The chemistry past 30 years, 40 years in Hyderabad is, evolved because of three institutes. The first one is Usman University, where we all are hailed from. The second one is IICT, where the international research, so many discoveries, so many patents happens. And I am very proud that, my senior from Usman University, now he is director and pushing the organization very well to improvise the chemistry skills, knowledge and patents, so that, the knowledge from India will gain the economy. Knowledge from India should, improve the employment to the people. So that's how DS Reddy Garu, Usman University is working. Apart from this, the Central University also. So these three institutions, including Kakatiya University, helping to improvise the knowledge of the students across the Telangana, and that is the reason the pharmaceutical industry in Telangana was topmost. The IDPL industry also helped. IDPL, a government organization earlier, now it is closed. But that also helped for the employment and motivating people to work towards chemistry. So now there are so many industries now, and because of the Shamirpet Genome Valley, more than 200 institutes are there, employment opportunities are there. People who are confused to select electorates, people who are in BSc and BPC, whether to choose biology, physics, maths, chemistry. As a chemistry person, I definitely recommend to select chemistry and work in the chemistry field. You will definitely get jobs, you will definitely get opportunities to prove yourself in your life from Hyderabad itself. And I thank for giving this opportunity, one and all. And I thank my CEO, Dr. Satyanarayana Chava, to sponsor for this program after requesting by the head of the department and Dr. Dayanand. And I thank one and all for giving this opportunity. Thank you very much.
Now, let us begin the session. I am very much delighted to invite Dr. D. Srinivas Reddy, our Chief Guest Director of CSIR IICT, and he also has additional charge of CSIR IIM, CSIR CDIR. He has 20 years of research experience, 3 years of administrative experience, and 7 years of experience in pharmaceutical industry. He has published more than 140 publications and he has 35 patents. Now I would like to request him to begin the session.
Okay. Good morning. Good morning. Good. Thank you. Sorry if there is some delay, technical glitch was there. Now we fixed it. Hey, what happened? అది ఆపేసి ఆన్ చేయండి. నేనే లైక్ హీట్ అయింది. Yeah, that is okay, but slides I have it. Okay, while they are setting up, I'll keep talking something. Hey, can you listen to me? Hello? Okay, good. So first of all, let me thank City College Chemistry Department team. In particular, Dr. Dayananda is, I know him for a long time. He is also my junior. I am happy to see some of my junior colleagues here. Dr. Manibhushan, Dr. Ravikumar Reddy, of course, some of the other friends also there. And I'm happy to be here. And thank you for inviting me to be part of this conference. And this is the second time I'm coming to this City College. So when I was doing my BSc, when I was like you student, I was doing my BSc in Sardar Patel College. So I used to have friends here. Particularly, I was part of, I see some NCC cadets there. So I used to be NCC. So that's how I know NCC cadets. So I used to have friends there. That time I used to come here, spend some time with them. So that's how I know about City College. And this is the second time I'm coming. This maybe after 20, 30 years. So just to tell you little bit about our institute, IICT, that is the Indian Institute of Chemical Technology. So we have several laboratories in India. So one of them is IICT. This comes under CSIR. So anybody know what is CSIR? CSIR. Can you raise your hands? Anybody know CSIR? So that is Council of Scientific and Industrial Research. That is one of the big organization that has several laboratories across India. So they work on chemicals, physical, engineering, information, biological. There are different labs are there. So that is one of the lab we have in IICT. In IICT there are other two labs. One is the CCMB. There, I'm sure you must be knowing, Center for Cellular and Molecular Biology. There is also NGRI. There are three labs. Total we have 3 CSIR labs in Hyderabad. So this one. Our lab works on chemical sciences. No. Huh? There is something. Yeah. So I had some slides. I wanted to show you some of the things of how chemistry is going to play a role in our life, and also some work what we do in our lab. That's what I wanted to show you, but it looks like still some technical glitch is there. No, most probably that heated egg. Okay, this is what our CSIR labs I was telling about. See, across India we have several labs. This is where our CCMB, IICT, NGRI are there. This IICT works on chemical sciences, right? So always the Prime Minister is our president for the society, and Vice President is always our Science and Technology Minister. And we have now a Director General, Dr. Kalai Selvi. She is the first woman Director General after 80 years of existence. So basically, I see many girls. It can be inspiration to become Director General. So that's something I wanted to tell about this one, right? So we work in different departments. Like we have process development and fluoroagrochemicals and polymers, organic chemistry, and also this biology, analytical department. There are various departments are there, but we mainly work on this healthcare, chemicals, energy, and environment. These are the four major areas we work, and this is what we have been working in our institute. So we have 150 plus scientists like me, and we have technical staff about 200 plus. We have about 450 PhD students. So they all, like you all, after completing master's program, they join with us. So they do research in various fields, right? So again, just to tell you a little bit about our previous speakers also talked about what is the role of chemistry in our daily life. I can tell you that chemistry is a central science for many sciences. Like look at this one, chemistry can connect with the biology, physics, astronomy, and medicines, and engineering sciences, geology, geophysics, everything. Like all sciences, they are linked with chemistry. So that's what knowing something about chemistry is worthwhile, because it provides an excellent platform to learn more about our universe where we are living, right? Hey, don't do that one. You don't do that one. Okay. So chemistry. So I don't have to tell you what where we require chemistry. Look at this one. Every part of our life is chemistry. Look at this one. Vehicles, what we are using today, in that all this fuel, polymers, whatever we use in our tires, they're all chemistry components are there. Similarly, so we all have smartphones, gadgets, laptops. There, all electronics also contain some of the chemistry component here. You look at this one, all these circuit boards, and this RAM, and also this LED screens. Wherever you see, our chemistry component is there. Similarly, even homes, like what we construct, the cement and carpets, paints, whatever we use in our home also, there are some chemistry components. There are polymers also part of chemistry, right? So that's what chemistry can give us: durability, safety, and also efficiency. And even in kitchen, right? Our mom cooks everyday food. So what happens is raw food is cooked to like cooked food, where it will become more digestible. So there is a chemical reaction is taking place. So particularly when you take this raw food, when you heat it with a solvent like water or oil, if you cook it, there is some reaction is taking place. That's how it is becoming cooked food. There is a chemical reaction is taking place. Similarly, all your cleaning utensils like dishwashers, laundry, body wash, even toilet cleaner, they also contain some of the chemical components. Particularly there is something called sodium NaOCl, that is a sodium hypochlorite, right? So madam was telling sodium chloride, that is salt, table salt, right? So that is also chemistry. So if you add another oxygen there, that becomes sodium NaOCl. That will be used for cleaning reagent. So that's somewhere, again, you look at this one. Ice cream, we all like ice creams. There is something called vanillin in this. Vanilla ice cream, you all know, right? Yes or no? So in that you have this vanillin, there is one chemical. This vanillin is coming from vanilla beans. That is a natural product, but what happens is the requirement is so huge, so we cannot meet the demand from the beans. So there is a way to make this compound. Now we synthesize this compound, we make ton scales, not kilos or quintals, it is tons. So that much requirement is required. So that is why synthesis is required, right? Similarly, we also look at onion, right? When you cut onions, you always you cry, not cry, but basically you get tears in your eyes. So there is one chemical responsible for that. That is the chemical is this particular compound, you look at this one, this is called propane thiol S oxide. That is something volatile compound that will give, so that will give, that basically that will interact with our sensory glands in our eyes, that will be reflecting for, that will be giving, that there is a reaction. That's how your tears comes. So there is always, there is some tips you get. So to avoid tears in our eyes, then our grandmas or some social media also you can see some tips. So you have to keep it in fridge and cut it. Another one is you can also cut onions inside water. Then you can avoid these tears, right? So what is happening is if you cool it down, this temperature of that particular onions or these chemicals present in the onion, that will reduce their volatility. So the boiling point will reduce, right? That's how you can reduce that volatility. Similarly, if you put in, so if you, you got other one? So that's why it is not coming out and it is not interacting with ice. So that's one of the, basically there are scientific basis for that. So that's where we can reduce our tears, right? Can you put this one? Maybe too close actually. Put there. Yes, technical problem. Finally solved it. And also this looking better, I think, this one. Okay, attention please. Yeah. See, I told about onion, right? There is also something about moods. Sometimes we are happy, sometimes we are unhappy, sometimes we cry, sometimes we are very joy mood. So these are all responsible again because of hormones levels. There are multiple hormones are there, but one particular one is this is called oxytocin. So that hormone level is that fluctuation also can lead to our mood swinging, like happy to like a sad moods. So that's and again responsible this one of the chemical, right? And similarly, so I don't have to tell about this all medicines. Whatever we use drugs today, they are all again made from chemicals. These are all chemicals. You can see this one. All this paracetamol, painkillers, histamine, antihistamines, antacids, antiseptics. These are all again made up of chemicals. Just imagine without chemistry, how many people can survive, right? So chemistry is everywhere, from starting from cooking food to cleaning vessels and all these drugs. Everywhere chemistry is important. So that's what I'm telling you. Something knowing about chemistry is worthwhile in our life. Even if you are not a chemistry student, better to know chemistry. So how chemistry can help us in our daily life, right? So just coming back to our institute. This is the Indian Institute of Chemical Technology. వాళ్ళ దాంట్లో HDML అది HDML స్మాల్ అయిపోయింది కదా ఉన్నాయి go back removable disc last first one. One chemical called hydrogen hydrate. This is one of the important chemical. So that's basically required for making agrochemicals and also making our drugs. That is one of the key starting material. So globally we need about 200,000 tons per annum, that chemicals. So in India requires about 10,000 tons per annum, right? So are you guys are listening here? Thank you. Okay. So this chemical, basically right now we import from outside, particularly from China we import a lot of this tons of material. But there is a need to make in India. So IICT last more than 10 years has been working on this project. Now developed a process at particular bench level and kilo level. Now collaborated with company called GCC in Gujarat, established a plant which can produce 10,000 tons per annum. So India requires about 20,000, but we are GCC established a plant about 10,000 tons per annum it can make. So that about 1.5 years ago, our Prime Minister inaugurated this facility dedicated to the nation, and I was part of that. One of the consignment, basically first production of commercial production came out. I was there flagging up that particular commercial of production. So that's basically great achievement from IICT side and CSR side, where we contributed towards Atmanirbhar Bharat, what we are talking about today, Make in India, because Make in India is very, very important so that you don't have to depend on other countries. That's where this is one of the chemical we are, with IICT help, we are able to make it. Right, just to tell you a little bit about this, we all went through these tough times of COVID, right? So many of you took vaccines, right? And drugs also. This that time when we got into trouble in March 2020, there was no medicines, no vaccines. So our IICT worked with CIPLA. Then in 3 months, like March we started. In July we produced a compound. There's a favipiravir. We developed a like process and transferred to CIPLA, and now CIPLA marketed the drug. So that's how now first drug came into the market in India, that favipiravir. Right. Similarly, we closely work with Bharat Biotech. So how many people took vaccine here? Can you raise your hand? Okay. Majority of you took vaccines, COVID vaccine, right? So one of the vaccine is Covaxin, that is developed by Bharat Biotech. So every vaccine has different ingredients in this one, adjuvants. So one of the adjuvants required is small micrograms, that is basically, it's called TLR78 agonist, one small component, that is chemical. So we didn't have at that time. So IICT, our scientists made that compound and gave it to Bharat Biotech, and Bharat Biotech like put it in that vaccine, and finally they released that vaccine. So here, whoever took vaccine here, so there is a small component of IICT is there. That is small micrograms of IICT compound what we made in IICT. So that's kind of satisfaction for IICT. Right? So then little bit about what we do in my research laboratory. So my research laboratory means every scientist has different research group. Students like you, after masters, they come and join with us. So we have research group in actually I started my career at NCL Pune, that is the National Chemical Laboratory Pune. So we work on this natural products synthesis and also this application oriented. Basically we make molecules and we make them useful chemicals. So that's how we do it. So why, like just to give you one example, these are natural products. Natural products means which are isolated from natural source. So we use those chemicals, we use this scaffolds and make drugs. That has been going on in the several decades. Particularly about 40 to 50% of the chemical or drugs what we get in the market today, medical shops, they are all linked with the natural products. Look at one example here. So this is called something called fluorescein. This is originally isolated from apple tree. So that is this is a compound here. So there is a later people made this compound called T1095, then converted into dapagliflozin and canagliflozin. These are the chemical structure. Those who are not very familiar with the chemical structures, what I'm telling you is this is a O glycoside, this is a C glycoside. This is a glucose linked with aromatic carbon, aromatic ring with O. That is O glycoside. So in our body, glycosidase enzymes are there that can cleave. But by the moment you put it C glycoside, that is stable to that O glycosidase. So that's how we developed some of these, like over the all companies and different academic group, they worked on this and they developed this compound. This is an anti-diabetic drug. During my work in industry at Advinas, along with Novartis collaboration, we invented a drug. This is called LIK066. That's called licogliflozin now. So now that compound is in phase 2 clinical trial. It has to go through clinical trials, testing. Like first you have to test in animals, then it has to go to healthy humans, then patients, then finally large scale we have to do this one. So this is something called phase 2 clinical trial we call it. So it is undergoing phase 2 clinical trials. So this is something, some contribution from our research group when I was working in industry, right? So these are some of the molecules. Again, I'm not going to go through the details, but these are the molecules, natural product we made in our research group when I was a scientist working at NCL Pune. right? Why do we make this compound? Because most of these compounds isolated from nature, they isolated in small amounts, very milligrams level. But whereas when we synthesize, we make in large scale. So that is the advantage. Then those large scale compound, we can use it for any purposes, any biological screening or any other purposes we can use it, right? So I'm not going to the detail, but just to tell you that how this works, just I'm giving you one example so that I took one simple example so that all undergraduates also can understand this one. Look at this molecule. This is called hunanamycin. So this is isolated from marine sources. Marine means marine bacteria. This was compound isolated. It was isolated 1 milligram of sample, just 1 milligram, right? That 1 milligram is not sufficient. So then you need to make good quantities of that material. There is another, look at this one, riboflavin. There is some structurally something related to this one. So this is well known in public domain, that is riboflavin. There's some structural similarities. So we believe that this is working through riboflavin synthase inhibition. There is one target in our, particularly in the body of bacteria. So where that has to synthesize riboflavin. So riboflavin in bacteria, they synthesize with the help of riboflavin synthase enzymes, whereas in our humans we get from food. So if you selectively block that riboflavin synthase, you can selectively kill the bacteria. So that is advantage, so that it will be safe for humans. That's what we are trying to do. We believed this one. So what we did is, first thing is we wanted to make that compound in good quantity. For any test, you need some quantity, right? So what we did is just to, for chemistry students, particularly who are little familiar with organic chemistry, so this is the starting material that we can make easily. If you take that corresponding diamine and oxalic acid or diethyl oxalate, if you cook it together, if you heat it together, you will get this solid comes out. So then you can do this alkylation using this prenyl bromide, you get this one monoalkylation with 42% yield. Then there is something called Friedel-Crafts alkylation. Right, I'm sure some of you must have heard this one, Friedel-Crafts alkylation. Aluminum chloride, Lewis acid we use, you get this tricyclic compound. Then you install this allyl bromide derivative, then you get to this compound. Then you functionalize this double bond into, basically you have to put 2 hydroxy groups, functional groups. Then you remove this protection group, you get this final compound. So this is called hunanamycin natural product. Originally it was isolated in less than 1 milligram, but whereas we made more than 80 milligrams within few weeks. That is something satisfying. Basically students who come from after training in masters, they come to my lab and they make these compounds. That's something interesting to us. So in addition to that, what we thought is this, when we look at the cost, we also need to make any compound in affordable price, less price. So if you look at the starting material cost, diamine, this is 25 grams, $450 US, right? Whereas riboflavin, 25 gram, only $32. So instead of making from this, we thought that we'll make from this particular compound. For that purpose, what we did is we took this riboflavin, very cheap, readily available anywhere, and we exposed to the sodium hydroxide, hydrogen peroxide to get this ring get cleaved, and you get to this material. Then you do prenylation, then finally Friedel-Crafts alkylation, you get there this natural product. This is something very interesting I want to tell younger students. See, look at this one. We took this riboflavin readily available, and we did not do any column or anything purification. Directly washings only can get about 48% yield of this final compound. So now look at this one. We can make now 10 grams of this compound in lab. So anybody, even summer trainees, they come to my lab. If I give them that benchtop material, they can make without much training. In 2 weeks, they can make this material. So that is something satisfying for our research group activity, right? So we also wanted to understand some mechanism to show that it is working through riboflavin synthase inhibition. I'll skip this one, but I'll show you what I'll do. Something else, something maybe interest to you also. Insect repellents, right? We have, right now we use mosquito repellents in our home, right? There are what we call all out, good night, all right. They're all there, right? We use in our daily life at home. That will be used for repelling mosquitoes. So their ingredients are these chemicals. So over the years they are all exposed. These mosquitoes are exposed to these chemicals. They are becoming resistant. There is a need for new chemical. So that's where, before that, I'll ask you one question. If somebody asks you a question, what is the world's deadliest animal? So you always look at big animals like crocodiles, elephants, tigers and all that. But today, world's deadliest animal is tiny mosquito. So mosquito kills 7,25,000 people every year. More than 7 lakh people get killed by mosquitoes, just small mosquito. So next deadliest animal is human, right? Human killing humans. They will kill about almost 5 lakh people every year. So then look at this one, other big animals, what we are thinking in the beginning. So they're all tiny, they get all the blame. They take only a small portion of overall contribution, right? So that's why if you can take out mosquitoes from the world, you can save this many people. That's where we're looking at. For that purpose, we have to make some compound. We identified a compound. So this is already known in the literature, but we identified another compound which is coming from Chinese medicinal plant. So we made this compound with using some of the synthesis what we know. Then we tested and we showed that at least there are 4 or 5 compounds which are better than original Nutcatone compound which repels mosquitoes. These are the mosquito species we used, right? So not only repels, it also kills mosquitoes. These are all like these chemicals what we made in our lab. I think in 2-3 minutes I had a video, but I will not be able to show because of PDF file. And here these compounds not only repels, it also kills, but at the same time it has to be safe for humans also. We tested in rats, so it is a safe molecule up to 2 grams, right? So right now this particular project is, we are working closely with industry and they are taking forward, right? I'll show you last story of this. There is about how chemist can help agriculture. So when we eat these grapes, draksha, so there you see white powder, right? You see or no white powder? So in white powder and also pomegranate, papaya, you see some white powder there. Actually, if you see that in a magnifying glass, they are actually mealybugs. They are all bugs. So the most of things we eat that time, right? So and also this can lead to loss of yields. Basically, you will not be able to produce enough yields if the crop is attacked by these mealybugs. There are different ways to do it, but one of the well-known one is insecticide soap. So insecticide if you use, but later of course there is always a problem with the environment, and also you can't export also if it is insecticidal residues are there. So there is a way to do it. There is something called new concept is coming, something called sex pheromones that can be used for taking care of this mealybugs. How do we use this one? So this females, particularly female mealybugs, they secrete pheromones. So these pheromones, what they use when they secrete, they get the pheromones get attracted by male mealybugs. So we use that pheromone secreted from female bugs. We put it in the trap here. The trap, that sticky sheet. So we put a small quantity of that material in the traps, then put it in the fields. Then all the male mealybugs, they come and they get attracted, they get trapped here. That is a sticky sheet. You take out that sheet and you burn it, few rounds, few cycles if you do, then you can eradicate all mealybugs, all male population. So in this process, there won't be any reproduction. There won't be any further growth of these mealybugs. This is one way to do it. This is very sensitive, very safe method. So what we did is, one of my students, she joined in my group at that time. We made both compounds, plus pheromones and minus pheromone, both compounds we made. And we collaborated with the US group, that is the Professor Miller group of University of California. We tested those compounds. We sent only 1 milligram because it's so powerful, this compound. This can attract up to 3 months. It can attract this male mealybugs. So we tested, we showed that this is a racemic compound, plus isomer, minus isomer. Basically in a racemic compound, you have plus isomer and minus isomer. So one is a mixed one, there is a racemic called mixed one. This 2 isomers is separated one, what we have. So we tested, we showed that one of the isomer is a plus isomer showed is much potent than racemic compound. And whereas minus compound did not show that much activity. So we published a paper, filed a patent. Then another group from New Zealand requested me to send some sample because they want to try it, because we published from US data. Then after that, then we sent another 1 milligram of both compounds to New Zealand. So there we tried again, it showed similar activity, in fact better activity than USA. You all know, right? If you visualize map, US is in somewhere else geographically and New Zealand is somewhere else. So both are different geographical location. Both places it is working very perfect with small amount of material what we made in our lab at NCL Pune. So then once we are successful, we thought that why can't we try it in our India? So in particularly Nashik area, vineyards is very famous for Draksha, these vineyards. So we thought we'll try it out, but we tried, but we were not that successful, only partial results came. There could be two reasons. One is we didn't know how to do that experiment, particularly this entomology. Entomology means like we work with insects. So other one is we don't know whether that same species is present in India or not, but whereas both in New Zealand and also in US, that same species is present at showing powerful results. So right now there is also request from Australia, we are going to try that one. Right? So what I try to show is we work in our research group, we work in application-oriented organic synthesis. We try to create some leads. It can be useful for either medicines, or it can be used for any crop chemicals, it can be used for many other purposes, any materials, or insecticides, or insect repellents. These are all the things we do it. And students who get trained in our group, at least they will work in that direction in application-oriented organic synthesis. Right? This is my research group. Again, these are all like you all after after MS Masters, they joined with me, and all these who are outside this box, they all completed their PhDs. Some of them are faculty, some of them are doing postdocs, and this is the current group, research group. So we are at IICT Hyderabad. So again, finally, I would like to thank my organizations who supported me and also collaborators, and also thank you all for your patience. Thank you. And sorry for that glitch. Maybe that computer also I should have got it with a different. Okay. Right. Thank you all. Dayanand.
Thank you, Anna. Thank you so much. Even after so much technical glitch that was there, okay, you carried on your lecture. Okay, that shows your ambition to convey the message to the young or budding scientists of this hall. This Azam Hall is a great hall that is present in this city college, having a long history. I thank very much our DS Reddy sir. One thing I always connect with DS Reddy, once DS Reddy means he changed the lives of the Kashmir Valley people. That is, Kashmir Valley people will cultivate a plant called as lavender. He created a purple revolution in that Kashmir Valley. He enhanced the income of the Kashmir Valley people tenfold by his research. Okay, that is a wonderful aspect. That is called a purple revolution. Recently in RD parade also, recent parade, Republic Day parade, CSIR had given one vehicle, that is, in that vehicle that CSIR had posted this purple revolution only in the RD parade. Okay, that vehicle contained, I thought it is a work. Okay, one of the proponents of this purple revolution is our great scientist. This scientist, D. Srinivas Reddy, is my senior, and I am grateful that he is the topmost third scientist in India. Topmost, that is among the top 3 scientists, he is one of the third most important scientists in India. He created many molecules as mentioned. Okay, I welcome Ravi Kumar Reddy also, come over to felicitate our senior. Okay, thank you very much. There are 2 questions from the lecture. Okay, one for the students only. Okay, 2 questions will be asked as mentioned earlier. Okay, that is for every lecture there will be 2 questions that will be asked. Okay, first question which. Okay, there are some instructions that are there. We are having 6 lectures are there, sessions are there. Okay, don't be upset and don't be hesitated. You just raise your hand once you know the answer. Okay, first one is, okay, just now sir had delivered his lecture. From that lecture only I am asking. Okay, what was the molecule that inhibits the riboflavin synthase enzyme of bacteria? Riboflavin synthase. Stand up. Come here. Very good. Muhammad Abdul Qayyum of second year had answered exactly correctly. He got the prize. Come here, come here. Another question is there. Okay. Next question. Very simple. It is that is in wine grapes or in many fruits there are white colored powder is there. What is the reason for that powder? Yes. Mealybugs. Very good. Come here. Come here. He is from final year. I request all my students to continue or to follow DS Reddy's speech and his message. Thank you.
Now I would like to request our chief guest, Dr. D. Srinivas Reddy, Director IICT, to receive the felicitation. I humbly request the dignitaries on the stage to felicitate our chief guest. I would now request our guest of honor, Professor C.S. Subramaniam, IITS, to receive the felicitation. I humbly request the dignitaries to felicitate our special chief guest. Now, I humbly request our guest of honor, Dr. K. Manibhushan, Vice President of Laurus Labs, to receive the felicitation. I humbly request the dignitaries to felicitate our special guest. I would now request Dr. Ravi Kumar from C1 Labs to receive the felicitation. I humbly request the dignitaries to felicitate Dr. Ravi Kumar. Now, I would like to request our principal, Dr. P. Balabhaskar, to receive the felicitation. I humbly request the dignitaries to felicitate our principal. Now I would like to request Dr. Eluri Yadaya, convener of the session, to receive the felicitation. I humbly request the dignitaries to felicitate our HOD of Chemistry Department. Now, I would like to request our dynamic Vice Principal, Dr. Viplav Dutt Shukla, to deliver vote of thanks.
On behalf of Department of Chemistry, Government City College, I extend my heartfelt thanks to each and every one of you. It has been absolutely privileged to witness such an enthusiastic gathering of intellectuals, scholars, researchers in innovative thought process of chemistry. First and foremost, our deepest gratitude goes to our esteemed chief guest, Dr. Srinivas Reddy, Director, IICT Garu, and guest of honor, Dr. Subramaniam Garu, Department of Chemistry, IIT Hyderabad, for taking time out of their busy schedule to grace this occasion and their invaluable insights and wisdom. Your enlightenment talks our passion for chemistry and extended our horizons, inspiring us to think beyond the boundaries. We would like to express our sincere appreciation to special guest Manibhushan Garu, Vice President Laurus Labs, for his contribution, have made immense value to this event and have undoubtedly enriched the knowledge of all the attendees. Furthermore, we extend our gratitude to our sponsors and partners whose generous support and collaboration have made this seminar possible. We would like to thank Telangana State Council of Higher Education, SSK Biosciences Private Limited, Laurus Labs for financial supporting us. For unwavering commitment to promoting scientific education and research is truly commendable, and we are immensely grateful for your continued patronage. It is a great privilege. I extend my deep gratitude to our beloved principal, Professor Balabhaskar sir, for presence and guidance have been instrumental in making this event truly memorable and successful. Your leadership and presence have set a positive tone for this entire event, inspire all of us to strive for the excellence. Last but not least, we express our heartfelt thanks to all the participants, delegates and attendants who graced this inaugural function, online and offline. We expect your valuable contribution to the vibrant discussions to be taking place in rest of the sessions. Your active participation will definitely make this seminar not just learning experience but also a memorable gathering. It is like-minded individuals passionate about advancing frontiers of chemistry. A special word of thanks due to all the organizing committee members and staff who meticulously planning and coordinating and hardworking behind the scenes ensuring smooth execution of this inaugural function. Special thanks to all NCC and NSS volunteers and all chemistry loving students, college teaching staff and non-teaching staff and press and media personnel. Your dedicated and commitment is instrumental making this event a really success. I once again thank everyone who played part in making this inaugural function great success. We look forward your continued support and participate in coming sessions. With this, I announce that the inaugural function has ended. Thank you, one and all. Now we are all breaking for 15 minutes. Students are requested to stay back in the auditorium. Snacks will be provided to all. And I request all guests and dignitaries to join refreshment in front of Magdum Hall. And the session will resume in 15 minutes. And Professor Subramaniam Garu's class will be continued. Thank you, one and all.
1 minute, please.
I would like to request our beloved and outstanding Vice Principal, Dr. Viplav Dutt Shukla, to receive the felicitation. I humbly request the dignitaries to felicitate our Vice Principal. Let us all join the session after the tea break. Thank you.
I don't think these days anybody talks about like I'm only chemist, I will do only these things, or I'm argument's sake, say for example, a physicist, I'm only going to do these things. So that means now this is the departmental building, the chemistry department building, and the whole idea is basically like it should have some relevance to the periodic table, whatever you can think of. And if you look at I'm sure somebody would have said this one, I'm not going to talk about these things, but at some point of time, there is something called green chemistry approaches. Green chemistry can be defined in several ways. Either you try to increase the atom economy, or you try to basically put a catalytic reaction, or you try to remove the hazardous compounds, and whichever way you can think of it. As I mentioned that, okay, so at some point, okay, somebody, see all these classical reactions, whatever you have seen, everyone will get impressed by either the selectivity or by the conversion, or by say for example the yield of the product, or in other words that you try to see that what is your atom economy in this course. If you think that you have done a reaction where like you don't have any byproduct, argument's sake, so then you can consider that as one of the clean reactions, because as I mentioned that you take benzene, you try to put one nascent oxygen, you get phenol. This reaction may not go, like even industrially this is not the reaction. But as on today, the industrial reaction, which is actually a pilot scale reaction, is like you take benzene, you take with nitrous oxide like N2O. Only one catalyst in the world works for this reaction. That catalyst is basically a iron based catalyst, iron GSM5. So whole story of it is that, okay, under what conditions you basically talk about, say for example, am I trying to look at the specificity of the reaction, or you look at that the selectivity of the reaction? The homogeneous plays an important role, because in the case of homogeneous reactions, I'm sure that you all in BSc or in MSc would have studied, even if you take argument's sake, okay, your Wilkinson catalyst. In Wilkinson catalyst, okay, so you probably would have looked at something called a Tolman loop, which essentially talks about actually a switching between a 16 to 18 electron, which is like an oxidative addition which is happening, and the reductive elimination which is happening. In both the cases, you are always looking at a 16 to 18 electron switching which is happening. Then some teacher would have explained to you based on the effective atomic number, EN rule, and which all of you would have studied it. That means if you really want to understand, okay, so if I look at like say for example the selectivity, the homogeneous catalyst plays a role. If you look at say for example, if you want some catalyst to be separated and recycled, so then you look at a different combination. And if you look at, like I explained to the other colleagues, actually if you do a small scale reaction and if you really want to do it up, then you have the three options, like you do a batch reaction, or you can go to a CSTR model, or you can go to a plug flow reactor kind of a model depending on the physical state of your reactant. If you want to understand the scaling up of it, one advantage you get from the heterogeneous side is that, okay, you can carry out reactions in a continuous phase manner, either in a plug flow or even a CSTR, even a fluidized way. So that means now each one has its own advantages. That means if you look at specificity or the selectivity of it, homogeneous plays a role. If you look at that, the homogeneous versus heterogeneous, if you look at two extremes of it, if you want to bridge the gap, why homogeneous is very reactive? The reason is that, okay, why it is highly selective? You can look at either in terms of the turnover number, or you can look at in terms of the turnover frequency, or even if you look at the site which is accessible to you. In the same context, you extend it little bit, an enzymatic reaction versus a homogeneous reaction. I'm sure that you all will get impressed with an enzymatic reaction, because enzymes have very, very high turnover numbers. The turnover numbers of the enzymes can be of the order of 10 to the power of 4, even more than that. Now, if you really want to look at actually homogeneous and versus heterogeneous on two extremes, each one standing here claims their own advantage, like homogeneous has advantage, at the same point heterogeneous has certain advantages. But if you look at the overall scenario of using the, say for example, if you want to use molecular oxygen as a reactant, it's not possible in many cases, because molecular oxygen, say for example, your cumene to cumene hydroperoxide typically can be carried out with molecular oxygen. That means you need to activate your catalyst, or you need to have an activated molecule, or you need to create a system where the activity can be carried out. So under those conditions, actually each one has its own advantage. So if I ask you one simple question, if you classically look at the textbook definitions, I think gold should not be a catalyst. The reason that gold is highly inert, the reason that gold reaction with oxygen, as a matter of fact, is a highly non-spontaneous reaction, But compared to that, you take iron reacting with oxygen is basically kind of a highly spontaneous reaction. But this is the definition which you find it in the textbook, saying that a bulk gold is very stable. That's the reason why I'm using the gold atom or the gold metallic gold, because it doesn't undergo any kind of an oxidation. You look at your electrochemical series. In the electrochemical series, it actually tells you the relative degree of oxidation ease of it. You take zinc versus iron, or you take zinc versus copper, it tells you that zinc undergoes oxidation, copper undergoes reduction. But if you take copper versus gold, okay, or copper versus silver, now silver undergoes reduction. Silver versus gold, gold undergoes reduction. So like that means this is all a relative degree of it. Now the earlier story of it is like this: you have a reactant molecule. Now you have a metal which you are taking it. Metal can do either oxidation or reduction. So depending on the substrate level, if the energy level of the metal is higher, then metal can transfer the electron, then you do something called a reduction. Or if it is system is like this one, then the substrate can transfer the electron to the metal, metal accepts electron. In this case, metal acts as an oxidizing agent in this case. But now the old story was, you keep, if a particular metal or a metal oxide, argument sake, okay, you have to change your substrate if it doesn't work. Argument sake, many of your teachers, students, in a particular case, if a reaction doesn't go, your teacher tells you either change the temperature or change the reactant concentration or change the catalyst concentration. Because we all know that kinetics are basically controlled either by the concentration or the temperature or the catalyst, whichever you are adding it. Now you can bring a new dimension that by tuning the size of it, you can in principle play with now the redox properties of it. That's what actually the system tells you. I'm sure those who are familiar, I'm sure everyone of you are familiar. If you look at the turnover frequency, like versus turnover numbers, whatever you are talking about, or even say for example turnover rate, all these things basically depends on the number of actually active sites or number of, say for example, in other words, the active sites. In the case of enzymes, the active sites are very unique. Only one substrate can reach that enzymatic site or the active site. But whereas when you come to the classical chemical reactions, the same palladium, you will use it in the Heck reactions. The same palladium, you will use it in the three-way catalyst. The same palladium, probably you can also use it for some kind of other hydrogenation reactions. Now in these cases, what are you doing it? You are actually trying to tune your substrate in such a way that if not this substrate, you try to go for some other one. But now the advantage you can find with the recent developments, either it can be a molecular, like say for example a click chemistry you can follow, or even try to design a catalyst based on a substrate. Now you are actually playing with the other side of it. That means the same catalyst, if you want to tune it, now there is a possibility you can do that one. That one you can understand. So all these things basically, in other words, actually you look at that, every reaction we think, I'm sure that your teacher would have impressed you with something called compensation effect. So that means like if you try to look at that the activation energy versus the say for example the pre-exponential factor, if you can find that a variety of metals, okay, so have some linear relationship, it essentially tells that, okay, so there is actually by choosing say for example this one, like a platinum, okay, you look at that the platinum based systems are more for electrocatalysts. Platinum based systems are known for non-organic applications. The reason that platinum is a good thing with hydrogen, platinum is a good thing with oxygen. But contrary, like you take palladium, most of the cases palladium, you can also use it for oxygen, you can also use it for hydro, that means for oxidation reduction reactions. I mean, like if you really want to understand actually how the temperature is going to affect the reaction rates, you look at that, the temperature dependence on the catalytic activity. If you try to look at the reaction rates on the y-axis versus the temperature 1/T on the x-axis, you come to this regime where now this is a thermodynamic limited, and if you look at that, this temperature range is mainly the kind of a mass transfer effect, and if you try to go to say for example higher temperatures, you get very high reaction rates, the reason that there are no mass transfer limitations attached to it. I mean, I'm sure I can impress you this one with another example. Okay, so I go back and forth. Okay, just one slide. If you look at that in a catalytic cycle, this is where your active site is present. You want to carry out a reaction basically by using, say, for example, a heterogeneous catalyst. Somewhere now in my reaction vessel, say, for example, you take your water bottle, inside you are dropping the catalyst, you put a catalyst, which is like a single pot reaction, you are doing it. Now you believe that there is a three-phase hydrogenation. Argument sake, a triple bonded molecule, like say, for example,
example, oh, you want to do a selective hydrogenation, then your substrate is in the form of a liquid, your catalyst is a solid, and your reactant hydrogen, one of the reactants is a gas. Now if a hydrogen has to approach your reactant molecule, it has to pass through all these cycles, like from somewhere an external mass transfer has to happen. From the surface of the catalyst, there has to be an internal diffusion. From there, the adsorption on the site, then the product formation. This is the whole catalytic cycle, you have it. Now, when somebody talks about turnover number, it means that our turnover frequency, like our turnover number, our frequency, what it means is that in the given time, okay, how many cycles are repeated? How many catalytic cycles you have completed? Starting from the first step to the final step, how many cycles you are repeated? Okay, so that's what essentially talks about, okay, the catalytic efficiency. Now when you classically look at like, okay, I'm talking about a system where you may have to, argument's sake, produce large quantities of the reactants. So in a way, like the production wise, if you look at in the, say for example, the waste whatever you are generating versus, say for example, the quantities which are produced, if you look at that, okay, say for example, the way you can prevent the pollution versus, say for example, you look at like the waste whatever you are going to produce in certain cases is going to be higher, whereas you look at even in the other systems like where you have the plastic recycling can be one of the things, renewable resources utilization, VOCs treatment or hydrogen storage, in all these aspects, like okay, essentially you have the other ways of the using catalyst can become handy for you. So the same reaction I'm going to compare with both homogeneous and heterogeneous in a way to understand how to overcome the shortcomings of the heterogeneous, at the same point how to get the good aspects of the homogeneous thing. Homogeneous is a highly selective system, when you has a good turnover numbers, whereas when you come to heterogeneous it's very easy to handle. It's a difficult thing for you basically to get specificity of the reaction. That you can try to realize. Say for example, if you look at traditional catalysis, even if you want to look at even couplings or known things, basically you look at that, the way you are trying to look at is that you have a palladium salt, palladium plus 2. I'm sure that there is actually somewhere it must be written, there is a, yeah, you look at that, there is a Tolman loop. If you classically look at, say for example, in the case of a homogeneous thing, what essentially you are doing it is that, okay, so you are adding, say for example, you are adding a palladium salt, okay, in situ, whatever you have, it is through that there is an oxidative addition and followed by, say, transmetallation you have it, and that is how, say for example, the product is going to switch. In other words, the ligand plays an important role in this case. But if you want to really look at a ligand-free kind of a reactions or even the solvent-free reactions kind of a thing, you can look at the other way you have it, like the same Tolman loop you have it. Case of say for example the heterogeneous one, you have a cluster, argument sake, to that, say for example, okay, so the loop you look at that, the palladium cluster, instead of an atom you have it. Now try to do, say for example, oxidative insertion, you get the same step. Then you look at that, the transmetallation. So then what you have is basically like some kind of reductive elimination. If you try to give a comparison between these two, you have a single site kind of a thing in your cluster, say for or in your complex, argument sake, you classically put instead of palladium, you can put your Wilkinson catalyst where rhodium comes here. But now if you look at the same way, I'm trying to talk about the palladium cluster with the miniaturized, okay, so or even decreasing size of the active material. Now in both the cases you can give a 1:1 comparison. So atom versus a cluster you have it, okay, say for example atom reacting with oxidative addition, leading to the oxidative addition, you are going to get the ligand basically. So the same way you have it here. Now the same sequence can be repeated easily, but the only thing you need to identify is that you need to have certain combinations. You need to also look at good kind of reactivities you need to think about, good kind of activated systems you need to think. If I have to just give you a bulk, okay, palladium, okay, so palladium oxide, even in the case of palladium oxide, palladium is present in +2. It may not be active for you. That means if you really want to understand why palladium is one big question, you have it like why palladium. So of course you can also do now with copper based systems, okay, but if practically look at this kind of coupling reactions are well known, especially in the case of the palladium kind of a thing. Let you talk about actually a Suzuki, or even let you talk about Heck reactions, or let you talk about glacier coupling, this kind of reactions, okay, where like you want to form a CC bond, palladium becomes handy for you because that the palladium has good reactivity for these systems under the conditions. conditions. Okay, so the reaction conditions. So the palladium favors the formation of, say for example, tetrahedral D10 system, and it also is known for D8 square planar complexes, you have it. So that means that the features afford palladium has a good electron donating and electron withdrawing capabilities, you have it. Depending on the loop you are looked at that, there is an oxidative addition, there is a reductive elimination. That means that essentially if you look at your basic inorganic chemistry, you are trying to play with the 18 electron to 16 electron kind of a switching in all these cases. Now you need to look at that, the advantages you have with a simple palladium which is actually a single site. Now the bridging the gap can be done that I have a bulk material which is not very reactive, whereas a single atom which is very reactive for you. Now the way you are trying to bridge the gap is that you try to decrease the size of it, so but you cannot get a single atom. In the case of, though people talk about single atom catalysis and heterogeneous catalysis, you cannot define a single atom in that case, but that means that you cannot really go to an atomic state or even in a complex where you have a single site present. Now heterogeneous is on one side, and okay, nano is bridging the gap between that, then you come to homogeneous, then you go to an enzyme. This is the sequence in which you can look at your turnover numbers which are going to essentially change. That means if these are the things which you know, these are the classical examples which are known in the literature. Say for example, then you look at that, the palladium may easily participate in catalytic cycle due to its closer lying of the homo-lumo energy gaps. In other words, palladium is a metal basically. Like you all know that in the case of a metal, you cannot define the homo-lumo. Only when a complex comes into the picture, now you look at that, the homo-lumo is going to come into the picture. So that means when you form a metal complex, you know that the bridging between or the energy gap between the homo and lumo is not going to be very high. So that means that's how one advantage you can look at. In a large scale synthesis, as I mentioned, if you want to do a reaction with a 0.1 gram taking to 10 gram scale or even to kg scale, it's not an easy thing to carry out. But if you look at the other side of the heterogeneous thing, a fluidized catalytic cracking which is happening at 10 to the power of 5 tons or 6 tons out of it, you look at argument's sake even propylene to propylene epoxy, propylene oxide. So these kind of reactions where the quantities produced are 10 to the power of 7 tons per annum. That means like you, under those conditions, you cannot always look at that, yeah, I carry out the reaction only under the homogeneous phase. That means you need to now look at, that means if you want to scale up the synthesis, actually now the flow chemistry is one thing which people talk about it, okay, because in a continuous flow synthesis, if you want to do a reaction, this is much easier for you. Or argument's sake, you look at that, you don't have to do any workup in the laboratory. Say argument's sake, you have a clean reaction, you just have to remove the solvent, you don't have to do a great, with a simple workup, you can isolate the product. These things are basically the advantages you have it. So I'm talking about actually the trying to understand the same reaction which is done in the laboratory with say for example a traditional homogeneous protocol. Exactly for the same reactions, now you try to look at that a classical homogeneous versus say for example the heterogeneous you develop. So that reactions which I'm talking about, because the whole advantage you have it when you decrease the size is that you look at that, this is how a catalyst particle look like, okay, maybe it's kind of an extrudate you have it, or some kind of actually a sphere you have it, because I'm sure you all know that when you want to do a continuous phase reaction, you need to ensure that a catalyst doesn't offer any back pressure, okay. So unless, okay, the catalyst smoothly allows, okay, the reactant to pass through, you cannot really expect the continuous flow synthesis. Even if you look at arguments like a plug flow reactor is nothing but you have a reactant coming, you have a product which is extracted continuously. So that means if you look at those kind of scenarios, your catalyst should not offer any pressure drop. At the same point of time, the catalyst should be highly active. The way you look at that, this is how a known catalyst bead you have it. Then when you try to minimize, okay, when you try to look at, okay, or when you magnify, if you have an atom like this one, so for example, if you try to decrease the size of it, then you can look at that each site is typically accessible to the reactants. How will you increase the reactivity of it? Say for example, I told you even in the initial time, if you look at that, the carbon dioxide activation is something which is very difficult for humankind as of now, because carbon dioxide is a highly stable molecule. But you look at the nature is doing the carbon dioxide activation, that is a classical textbook reaction, a photosynthesis, where two sluggish molecules are reactive in this case. Your carbon dioxide reacting with water molecule, that means that the light reaction versus the dark reaction you take it, carbon dioxide even goes in dark reaction. That means carbon dioxide activation is much more difficult compared to the water activation, because water demands lesser energy compared to carbon dioxide. But nature does this reaction. Nature does it in a very complex way. Even if you want to do artificial photosynthesis in the laboratory, you don't get even glucose molecule. You will be happy if you end up with only a methane molecule, as a matter of fact. So the glucose production, the way it is done, nature does it in a very complex way. Like, okay, you cannot say that I'm a chemist, I do this reaction. You cannot say that I'm a biologist, I will do only this reaction. That means that you need to have the bridge of everyone. You need to understand the whole chemistry, how it goes, whole science, how it goes. So that way, where you can have an advantage. I'm sure somebody would have said this one, or I'm sure many of you are aware. The way when you decrease the size of it, you have several advantages. Those advantages are very simple to make it. First and foremost, when you talk about heterogeneous reaction, you need to have more surface. Argument's sake, we all believe that hydrogen reacts with oxygen in a beautiful way because this reaction it's an exothermic reaction. But I give you 1 liter of hydrogen, 1 liter of oxygen, okay, make it stoichiometric, 2 liters of hydrogen, 1 liter of oxygen. Even after 1 century, you don't get a single drop of water molecule. The reaction has a huge activation barrier. You never form water molecule. Then the way you do it, like, okay, you put a copper catalyst even at 400 degrees centigrade or 350 degrees centigrade, you see beautiful water formation, and you will be surprised that this is the whole crux of your fuel cell reaction. In a fuel cell, your H2 has to be activated, O2 has to be reduced, you need to form water, okay, so that's what essentially the final product you have it. But even in this case, we don't have any catalyst which can effectively work without any problem. So platinum works, but I think there is no substitute to platinum. That means you need to, when you change the size of it, when you cut down the size of it, earlier days you were like, when I was a student, if a reaction doesn't go, the idea was that maybe you increase the catalyst amount. Okay, or increase the temperature. But now the story is that you don't have to increase it, you decrease the catalyst. At the same point of time, you don't take it in the form of bulk, you just try to grind it very, very well. I just give you one classical example. This is nothing new for all of us. Say for example, today you go back home and you tell your mother that, yeah, it was like heavy load of lot of lectures, I have a headache or I have a fever. Your mother immediately tells that don't eat food, but she will be unhappy if you don't eat anything. She will give you some kind of a khichdi or idli, this kind of things, because your body doesn't have the energy to digest that regular rice grains. At the same point of time, a smashed food or mashed one, basically, like that means decreasing particle size makes it more active for you. I mean, your body doesn't have the energy to digest a large grain size, whereas when you cut down the size of the particle or cut down the size of it, you can digest it easily. Or cooked food, okay, so idli is steam heated one. So that's the things which you can follow. Colloidal chemistry is nothing new for us. The only thing is, this is even smaller than the colloid, so the reactivity is going to change basically when you decrease the size of it. That means when you cut down from bulk, okay, to decrease the size of it, you are actually trying to play with the redox properties. I look at chemistry only from the redox point of view. Any reaction, though you talk about an oxidation, there has to be an equivalent reduction, otherwise the electron flow cannot happen. That's exactly the same way you look at, like, you increase the surface area, you have, say for example, typical redox potentials you are trying to vary. So this is actually a confusing term to each of you. You look at that, if you have a simple molecule, you call it like a HOMO and LUMO. Then all HOMOs will transform to something called a valency band. All LUMOs will transform to a conduction band. Then valency band is a measure of the oxidation, conduction band is a measure of the reduction. That means if you simply look at that in one simple concept, you can understand the HOMO-LUMO, which everyone talks about as a molecule. When you come to a band gap like a semiconductor physics or semiconductor chemistry, you look at like, okay, especially in the case of your photocatalysis or photoelectrochemical cells, where you need to always play with your oxidation reduction potentials. Then you come to basically nothing but an oxidation reduction measure can be explained based on the size of the particle. Of course, the surface area increases. Then you also have the preferred morphology orientation. If you look at the advantage of nanocatalysis you can get, so you can improve the efficiency of the reaction. You can reduce, basically there is also a possibility of activating carbon dioxide directly to carbon dioxide reduction you can do. But whereas when you come to the catalyst point of view or from the organic synthesis point of view, you can improve, you can attain basically the atom economy, because like say molecular oxygen activation, molecular oxygen is a paramagnetic which is known to all of us, which is highly reactive system. But you look at that, the moment any carbon you try to put with molecular oxygen, you will always end up with a deep oxidation. Many times you end up with a deep oxidation. That's the reason why benzene to phenol cannot be done with a simple molecular oxygen. Benzene to phenol is basically done with a nitrous oxide molecule, whereas N2O you have it. What you need is a nascent oxygen. The latest now, the pilot scale synthesis of phenol now goes with not the cumene route, but you can directly do with take benzene and try to put with the N2O and try to put iron oxide catalyst, iron GSM5 catalyst, that gives you only phenol and nitrogen. Now you look at the atom economy in this case, I think the selectivity or these things will be advantageous. Now that way you can look at, so that means that, but you may also ask a question, sir, atom is not stable. Even if you say a very small particle, how will I stabilize it? In the case of enzyme, argument's sake, you look at even in the case of a chlorophyll, basically. The active center is always protected with a porphyrin rings or even some kind of pyrrole rings, basically, or pyrrole moieties. If you really want to do that one, you need to protect your molecule. That's why Wilkinson catalyst, where rhodium is protected, now rhodium single atom, basically it is going to be stable in that complex. Even if you look at hydroformylation, your cobalt catalyst, it's going to be there. So that means now you want to design that one. That means you need to disperse your material, you need to stabilize your material. For that you need to have something like a support. A metal cannot be stable, any decreasing size even not stable. One may simply ask a question, sir, metal is already an activated state. You don't get metal directly outside, you get only in the form of a metal oxide. This is an ore. From the metal oxide you are trying to extract a metal. That means that a metal oxide reduction to a metal, this is where even Tata Steel does, this is where even everyone does, a metal oxide has to be reduced. Okay, for that you are using a reducing agent. Now, once you convert it to a metal, metal is already an activated molecule. Any activated system is not stable. Now you need to know how to stabilize it. That means what? The moment you cut down the size, you need to know how to stabilize this one. For that, you need to play with basically the kind of support you need to worry about it. So a simple way in the laboratory to take a metal nanoparticle, it's not a difficult thing. Take silver nitrate, just put some amount of sodium citrate, just heat it in front of your eyes, you will start forming the metal nitrate, the metal, which is a silver in this case. But the same way you want to do, you can also do with something like basically a reverse microemulsion. That means in a controlled way, if you want to make the particle, there are ways to make it in the literature, because if you cut down the size, the activity varies. In the case of actually homogeneous catalyst, the size of the metal center is a known thing basically, like okay, or the active center is a known thing. Now if you want to repeat it or reproduce it, you need to make exactly the same particle size. If somebody talks about 8 nanometer, you need to make exactly an 8 nanometer particle size. Further, there are ways you can make it. This is just like a reverse microemulsion, which you can study it in the literature. The sequential way of doing it is I have listed here. There is a possibility you can make a metal nanoparticle. You can also use them. So then you look at that, what are the features I am looking at now? I want to look at the activity, I want to look at the selectivity. So then you need to also worry about something like your lifetime, and also the say for example, you need to worry about the cost, all these considerations. I just give one simple example. This is a simple, say for example, biphenyl synthesis. If you really want to go, you look at a simple, like say for example, you have a boronic acid, simple Suzuki reactions you are talking about. Say for example, you have idoarenes, okay, so for example, idobenzene, you have it. Then you try to put it, you get exactly the same thing what you can do. In the literature, you can find umpteen examples with the same reaction. You simply do a palladium based system, okay, say for example, simple palladium state you can put it. But now the palladium and carbon is something which you want, then you try to vary now this carbon support, because I want to ensure that the palladium nanoparticle, whatever I have put it, it is stable for you. Now you look at the way you are trying to play with, this is just like an optimization. Then you can also say for example, you can extend this one, the scope of the reaction, and exactly the same way the scales of around 80 to 90% yields you can get it this one. This exactly now you can repeat it, the same reaction whatever you have done in a small scale level, you can do in the laboratory easily to say for example 2 grams level, okay, so or a gram level you can do it, okay, so with this one. The advantage you have it is you need an yellow flag condition, maybe in 100-120 degrees centigrade you are doing it, and you have a good catalyst. Sometimes you can use a magnetic catalyst where now the catalyst can be easily separated and you can redo the reaction. Only thing is that this is done actually in a controlled way, like in the sense that you are done it under homogeneous conditions, and you can also do under heterogeneous. Now the scope you can extend it, okay, so it gives a view around good yields of around 85 to 90%. So that means that, okay, so this is something which you can extend. Even you can take argument sake, say for example benzofuran's. If you really want to extend it to benzopherones, you can do that one. That means there is no limitation for you. Only thing you need to know how to make the nanoparticle. Only also not enough if you, nanoparticle making is very easy nowadays. Okay, as I've shown you, you just go to silver nitrate and try to put a reducing agent in front of your eyes. Okay, there is a reduction. You can feel the formation of the particle. But somebody does the reaction at 40 degrees centigrade, another person did at 50 degrees, another person at 60 degrees. You see the difference because the color change tells you that the particle size formed is different. Now you need to make it in a controlled way. That's what I told you, that in a controlled way you can make it. Then you look at that, the recovery of the catalyst, which is, just 5 minutes, okay then? 2-3 minutes. Okay, you need to look at it in a controlled way. Say for example, the catalyst can be effectively recycled. You can use it. So now one in the literature, somebody talks about palladium and carbon. At one point of time, activated carbon was the only carbon. Now you look at several carbons you have it. Okay, which carbon you want to use it? Is it a single walled? Is it a multi-walled? Is it a graphene oxide? What advantages you can find? I think these things you can realize easily. The way to make it is very simple. You need to just make it like you need to have some kind of a reducing agent where you get a palladium oxide, then you try to disperse it on some carbons, okay, so that you can carry out. Now you look at that, I'm actually acrylates, okay, so another thing which I'm showing it now. The same, if you really want to look at that, okay, so if argument's sake you need to optimize the condition. So if you do actually this homogeneous versus the heterogeneous, you look at the differences you have it in this case. Typically you look at the turnover numbers I'm referring to, and the turnover numbers in this case are basically like around 400, okay, something like. This is something, it's a reasonable thing. Then you can extend the scope to a variety of things, okay, say for example, you can produce in a beautiful way. Just like, okay, so for the paucity of time, I just conclude by making some statements. It is ideally speaking that I need an enzyme because enzyme does only one reaction. Enzyme does the reaction in a very beautiful way with a very high turnover numbers. I cannot always get an enzyme, so I have a homogeneous thing which is highly specific, and the homogeneous thing gives an advantage that the reactivity is very good and the specificity is very good. And if I want to replace that one, now I cannot straight away go for a replacement because I need to match the reactivity of my homogeneous system. That means that the turnover frequencies or the reactivities I need to match without compromising the yield of it. That means that your yield or the reactivity should be also good. At the same point, you need to also get a good yields which are now known for homogeneous things. So we made an attempt, okay, so to try to replace the homogeneous for the same classical reactions in the laboratory by following the same protocols. Now you can actually say that, yeah, if you control the palladium size of this much, in a way like we sequentially varied from 2 nanometers to 40 nanometer palladium size, now you can understand that, okay, in the literature you find a variety of examples. One talks about 40 nanometers, another one talks about 100, another one talks about 5. Unfortunately, the definition of nano is like anything less than 100 nanometers is really good for me. But a 20 nanometer particle behaves totally different to a 10 nanometer particle. That means you need to know how to make in a precise way. Two metals of the same size behave differently. The same metal of different sizes behave differently. The same metal of the same size but the morphology is different, they behave differently. All these things you need to understand. Okay, if you know, then the reactivity of the homogeneous thing, the advantages of the homogeneous will not be compromised, and you can make a system like that. So that's what I actually wanted to convey. There are many things which you can do. I mean, since this is not an ego boosting session, I don't want to impress you what I am doing in my lab, but all of you are now the budding students. Many things can be done. Now there are no boundaries in chemistry, like there are no boundaries in science as a matter of fact, okay, because as I said, we extend help to many MTech departments, okay, so like we offer out and out energy science and technology 60% of the course. EV technology we offer, okay, so everything, like Dr. DS said that the time you get up to time you sleep, okay, you have, chemistry is with you always. It's only for us to make the right use of it and to understand why it is, because the science is something which you need to understand to put that in a logical way. And I'll be happy if all of you pay attention to the rest of the lectures so that you understand what actually can be done. One person cannot do everything, but the understanding is something which you keep with you. And I'll be happy if this seminar will be one step ahead in improving the knowledge in you. And I'm thankful to all the organizers, and many thanks to all the students. I know that I'm in between you and the lunch, I think, but I think I'll be happy to take up any questions that you may have. So thank you very much. Ask, I will repeat your question. Correct, correct. No, not mathematically. I am an experimentalist. I will tell you experimental how to do it. Sir's question is that, okay, a monometallic sometimes will be reactive, but it gets poisoned very easily. In a way, if you don't want the metal to get poisoned, a simple way, you look at your classical physical chemistry, electrochemical series tells you that. When you have, so argument's sake, when I want to do palladium, like unfortunately we have seen that the reactivity of palladium maybe after 7 hours it's going to decrease. Then you look at the way palladium get easily poisoned. If you really want to protect your palladium, the slide basically you have it, it's going to tell you how to do this one. If you have only palladium, that means the reactivity of palladium is something which is governed now, it will get poisoned. But now you try to combine with another metal, mainly you look at like now I combine this with the copper. If you take zinc and copper, you know that zinc undergoes oxidation, copper undergoes reduction. But now I want to protect my palladium, then what I have to do, I have to push palladium to be a reducing thing, rather than say if I combine this with copper, now copper undergoes oxidation, palladium undergoes, now palladium will be protected now. So that's how like the palladium nanoparticle, as I said, nanoparticle is highly reactive. In the case of a homogeneous catalyst, we have a protective environment so that now the palladium center is our active center of rhodium is protected in this case. Now in a heterogeneous way, if you want to protect it, you need to combine with a suitable metal so that your palladium should remain as palladium 0 in the case. So that's what actually we have done this one. As I mentioned, okay, so that's what, when you combine this one, earlier we have done the same reaction only with palladium oxide. Now what we did was, we took actually copper, but I cannot put directly copper. What I did was, I took another form of the copper in the form of a spinel, which is solid. On that now I decorated palladium the way it was shown. Now the copper present in my big matrix is going to protect my small particle of palladium. That's how like you don't get compromised it. Otherwise turnover number can be expressed at any point of time. Turnover frequency you are going to average the time. Everyone reports like only the highest turnover number I get it. Highest means that it's very clear that you have a reactivity loss at one point of time. So to protect it, you need to ensure that your palladium 0 is palladium 0. Because any metal which is going to be highly reactive, what reactivity? It goes to palladium 2. The way palladium to activated in homogeneous is different to heterogeneous because I have already a stable palladium oxide with me. Then I don't have a big ligand effect in this case. Whereas in the case of homogeneous, the ligand plays an important role in kind of a transmetallation reaction. Now the way it is protected in this case, now the way zinc and copper, zinc is oxidizing, copper is reduced, copper undergoes reduction. Now copper and the palladium have taken it. Copper undergoes oxidation, palladium undergoes reduction. That means if there is a possibility of palladium undergoing oxidation, now copper comes handy, it says that I will undergo oxidation, but copper remains as palladium remains as palladium 0. That's the effect we have followed. No, we are all basically teachers. Now we teach all BTech students. Okay, so please. Since madam is standing behind me, I thought my time is over. That's the reason why I concluded. Go ahead. Sir, actually, see that in the case of, say, for example, in heterogeneous case, there are two effects to understand. One is called a spillover effect. That means that you have put palladium on carbon. Now, first and foremost, the hydrogenation, when you are doing it, now you need to have a lot of uptake of hydrogen. Now, metal is an active center for hydrogen, but the spillover has to happen, so that means your matrix has to protect now basically. So that means it has to stabilize your metal nanoparticle. That for that, you have a variety of carbons. Other palladium carbon is a commercial catalyst. So now in a way, if you want to understand it, you look at now the electronic properties of the, say for example, the support. You take a single wall, you take take a multi-walled, you take a graphene oxide, you take reduced graphene oxide. In a way, you also see the whole PhD thesis of this one. We try to only look at the same palladium particle. We always try to vary now the support to really understand where in which support is giving you the best activity of it. Okay, so this is actually this is a combined thesis with Professor Satyanarayan and myself. So Satyanarayan is a hardcore organic chemistry faculty. Like I am a hardcore heterogeneous catalysis person. Then we took a joint student, and the student did everything in a homogeneous way with me. Then you look at that, the beautiful way of understanding it is that how support stabilizes the metal center basically. For that you need to have an optimal interaction. One thing just to add to what sir said, if your support is going to be very, very stable, then your palladium is, see, you don't want, you look at the catalytic cycle. Your catalyst particle should not hold the reactant too long. At the same point of time, it should not also have zero reactivity. It should attract to reactant molecule, it should convert it to product, it should desorb the product. If it doesn't desorb the product, it becomes a chemical reaction. So that means active center is blocked now. So how quick the cycle is going to complete, that is basically called the turnover number. So classical enzymes will have a turnover number of 10 to the power of 4, 10 to the power of 5. But if you look at a chemical reaction, it is a stoichiometric reaction. Now the way you look at a bulk material whose turnover number may be 100, 150, homogeneous has around 300 or 400. Now you want to bridge the gap, you can do this one with the nanocatalysis. That's the idea we put forward, and that's what the student has done it. Okay, so that's what I wanted to convey. Fine.
Please ask the student. Please ask. Oh, student. The student, not participant. Okay, we be here. Okay. Nice lecture by our Professor Subramaniam sir. Sir had explained all those aspects of nanotechnology. Okay, we are very sluggish, that is CO2 and H2O, but sir had made us to be more active by increasing our energy. Okay, two questions, my dear students, which I had to ask. One is, what is the turnover number of enzymatic reactions? Okay, sir had explained homogeneous. Okay, what is the turnover number of enzymatic reactions? Can anybody answer that one? Okay, shall I go to the next question is, in a given time, the number of catalytic reaction cycles that are completed is called as, is generally defined. The simple definition, sir was the main core of his lecture is only this point. Okay, then in a given time, the number of catalytic reaction cycles that are completed is called as.
I carry the books with me now.
Okay, another one. How to prepare silver nanocatalyst? We had to add silver nitrate plus some X. What is that X called as? Very good. Sodium citrate. Very good. Last question. Why? Because we had to answer two questions. There are, okay. Palladium is mostly used as a catalyst because of its special character. Where we cannot use copper, we cannot go for rhodium or ruthenium, but palladium is having a special characteristic property with respect to the energy levels, modular orbitals. Huh? What's that? Okay, among two reactions, heterogeneous and homogeneous, which is highly selective? Okay.
Multiple answers we get.
Multiple answers.
You ask one more question. Okay.
How to increase the catalytic property of palladium? In Heck coupling, how to increase? Or what do you understand? Okay, in order to increase the effectiveness of the catalyst, what we had to do? Especially metal catalysts.
Okay, I ask a simple question. What is the atomic number of palladium? This is the toughest question, correct? What is the atomic number of palladium? You tell me the group in which the palladium present. Okay, so yes, yes. Amma? 40. 46. Oh, that's what your Google said, or you know it? Okay, come, like, okay, good, good. It's 46. Miss. Come. Come. Like, ఎవరు?
A very good afternoon to all. I take this opportunity to thank Professor C.H. Subramaniam sir for his wonderful lecture. His lecture connects and encourages the students towards the chemistry. Sir has started with the preparation of simple molecules like phenol, methanol from methane, and hydrogen peroxide from water, and he explained BTech programming in industrial chemistry, green chemistry in several ways, selectivity or conversion, atom and atom economy. Professor had explained metal redox reactions, and he has explained very clearly. Sir had explained about the homogeneous and heterogeneous reactions. Sir have mentioned in his talk palladium catalyst in CC coupling reactions like Suzuki coupling reactions and Heck olefination reactions, and sir has concluded with how homogeneous catalysis reactions are advantageous over the heterogeneous reactions. Really, here the faculty and students benefited by your lecture, sir. Thank you very much.
So now we are going to stop the session. So next we are going for lunch. So please, all delegates who were registered, please go to the our principal room. IQS room is there, and NSS volunteers are there. They will guide to you. And students, please be stay here only. We will give the token, and we arranged lunch for you in second floor at chemistry department. Okay? First, the delegates will move. Then we will issue the tokens. Then you go and take the lunch. Thank you. Thank you very much. Next session will be start at 2:30. 2:30, please. After taking lunch, next session will be start here only. Please gather at 2:30. Thank you.
Usmania University in the genetics, and PhD also from Usmania University in the genetics only. Sir has a lot of experience as postdoctoral fellow in most reputed institutions like Directorate of Rice Research, Hyderabad, Institute National Dilla, Richard J. Agronomic, France University, like that. And sir has honored with the various honors and awards. He is the member of the national and international societies, and sir has one book publication and 6 chapters in a book, and about 45 research papers, and sir is the reviewer for the 10 reputed journals, and there are 6 students pursuing PhD, and 4 students have completed their PhD under the supervision of the Rajendra Rao sir. Sir projects has funded by the DBT, ICMR, NIH, UKRI, and interested area of research is impact of nutrition on non-communicable diseases, genetic and epigenetic approach. With this brief introduction, I welcome sir to give the lecture.
Okay, namaskar. Let's start. This umbrella can be side lo pettukocha. Unda collar unda. Okay, good afternoon. After lunch, no, bit dull because no, this is on diet, nutrition. Say loudly, bit loudly. Good afternoon. That is the enthusiasm it shows. Yes, after lunch also we can listen carefully the boring lecture which I am going to deliver. You have to see that no, lectures are 2 types, very active and the boring. That boring we are going to see now. You like it? Okay. Whatever it is there, then we have to enjoy the both. So first I would like to hello, it's okay, right? So first I would like to thank the organizers for inviting me, choosing me for this today's international symposium on chemistry, the emerging international horizons in the chemical sciences. But I am not a chemical scientist, but I am a nutrition scientist. How I am fit into this? Mobile, it is okay? Oh. Hello. Right. How I am fit into this? Big question, no? Nutrition, diet, after lunch, Rajendra sir, sleepy. So let us see that how best I am fit into this one. Thanks to Yadaya sir. He will not leave me because no, I was in the on duty in Rajasthan. He said, he called me, you have to give a talk. Anna, I am coming on 28th. He said, no, you have to be here on 19th. I preponed my trip and I came to Hyderabad. Thank you, Anna, for choosing me. So that is the confidence levels where we can build what we need for any talk, whatever we give. So whatever the work we are doing, we need that type of knowledge. So in that same direction we will go. We are what we eat. We are. I am like this. What I am eating? I am eating food. I am eating fruits. I am eating vegetables. I am eating biryani. I am eating chemicals. So chemicals almost 99.9% people, everyone will take one or the other medicine. If not, we can take that Mysore bhaji or bonda, in that no chemical is there, that is called maida. We are eating the chemicals. So that no, what chemical is doing with the nutrient. Can we discuss the diet and nutrient and drug interactions? That is the one idea to go for it. Or shall I speak about the general things, I thought. So instead of going for the science, everyone is speaking the science. I saw all the topics, but I want to go in a general way. I speak about my work. So that is called diet gene interaction and obesity. How the people are developing the obesity. So what is the reason behind it? So how to overcome the problem. Is it okay? Here, yeah. Healthy diets. Everyone want healthy diet. So arising either by the tradition or design. We are having a tradition. Every home, we are having a one system to follow, one system to eat. Yes, in our tradition, we eat this. If you go to the Rayalaseema side, of course in Telangana area also we used to have the millets at childhood. Now it changed, total transformation. We are eating the rice. So when I was young, I used to have the bajra, so jowar, and the wheat is less, of course, ragi. These are the things. Now the transition has come. So from there to here. So everywhere, every place. I had been to the Rajasthan. Their food habits are different. I had been to the Arunachal Pradesh. Their food habits are different. I had been to Kerala. Their food habits are different. Everywhere we are eating their local foods. That's what, no, when they are started eating the local food, there is no problem at all. When there is started adapting the other things, no, the problem started. So that can we, this a healthy tradition and the design share many common features and the generally align with the WHO global action. Here in India, wherever you go in Bharat, we can see that idli we eat. In Kerala, they eat the puttu. In Gujarat, they eat the dokla. These are all fermented. So Orissa, if you go, so the pagal they eat, that is also fermented. So these are the fermented foods we are eating more here. So that that traditionally we are having, where we are adapting the prevention and control of the non-communicable disease, which is in our hands. Non-communicable disease is in our hands. Communicable disease, which is coming by bacteria or virus or fungus or something, but non-communicable is in our hands. So we can prevent all this. In comparison with a Western diet, these healthier alternatives are higher in plant-based foods. Western diets, now we are no pizza burger is a fashion to eat. No one knows that what is the composition of the pizza burger, what is the base of the pizza, we don't know. But we know whatever we are eating at home. So dal, so pappu, charu, kuragayalu, then we know everything is the composition. Whereas do we know that what we are eating in the pizza, in the burger? No, we never read, we never understand. So that the Western style of is different from the our local this thing, including the fresh fruits and vegetables, whole grains, legumes, seeds and nuts, and lower in the animal based foods. Here most of the people they eat the vegetarian food. So there is a difference is there, some commonality is there, some differences are there. These differences mismatches where it is taking us, we'll see. Balanced diet. The balanced diet contains an adequate amount of all nutrients required by the body to grow. We need the proper growth of the person. So then remain the healthy and be disease free. So we should be healthy. During corona time, all we feared, people are dying. Who are having the immunity, it is not a problem. We can defeat the virus. Who are not having the immunity, that is the problem. Then when we started giving the vaccine, then we started from the elders to the down, because children are having the more immunity compared to the elderly people. So that this is the where we can see that diet is giving to give the immunity. In addition, healthy balanced diet provides the necessary energy requirements, protects against vitamin, minerals and other nutrient deficiencies and builds up the immunity. So when we are not taking the holistic, the deficiencies are common. This is wherever I go, I say everyone likes to be that fresh morning when you wake up. I want to be healthy, fresh and good. My life should be good. All we are dreaming. How it is possible? Is it possible? Be cheerful. Like that, no? The plants, the nature is going. It will not go. That is the rainy season or the summer or the winter season. Every plant is having its own, bloom of these flowers, it will give the fragrance, it never stop. It never go against the nature, but we go against the nature. There the problem, mismatch will come. Here also you can see, what are you seeing here? The flowers. Flowers are colorful flowers. The colorful flowers are flowers, healthy flowers. This is a grass flower. When it comes, now first rain comes, second day, third day it will come out, fourth day it will give, starting the budding, fifth or sixth day it will give a flower, and sixth or seventh day it will die. Nice, no? By using that grass flower, we enjoy a lot. Wow, nicely. We want to be like that. But we can see these are all in mud. Buradalla untai, we can see somewhere on hill area and all the things. So you want to be keep your health. The diet is the proper important thing. So here, how many of you are eating the nutrition, proper food, I don't know. But every day, Hyderabad, we are blessed, we are in Hyderabad. Very cheapest food is the fruit. On the roadside, we can say they are selling lot of fruits and vegetables. But whenever we take, that will come. So are we taking? Then question yourself, answer yourself. Try to eat the fruits and vegetables. This is the nutrition institution we give. We educate you people to eat the seasonal fruits and vegetables, where that can give the health to you. Here then come to the chemistry. The chemistry and nutrition. Then what is the chemistry is there in that? So people are isolating the secondary metabolite from the plants. Secondary metabolite is the chemistry. People are isolating the Ayurvedic compounds from the plants. So the compound is coming. When we are purifying, that is a chemical. Otherwise, whatever we are eating a chemical. But it is a mix of everything. When the chemistry will come, that is a purified one. So that is a simple difference. Here in this everyday life, we are eating many things. So then what is there in that? Whatever the plant you take, in that carbon, hydrogen, oxygen, nitrogen. That is the composition. Those we are consuming here. So why we have to consume all the things? Here then we can see drug products are there. Here in pharmaceuticals and pharmacokinetics. When it is entering into the body, we can read all the things. What is happening, all the metabolism and all. And the nutrients is other side, the food and nutrition. That is also the same. When you take the drug, we read the same. When you take the food also, we can read the same. Both are the same. One is the purified compound, one is the whatever the nature it is giving. So both ways we can see, so that the chemistry and the biology is linked. So that in this, what we are trying to understand is here how it is changing our physiology, our body metabolism. Metabolism is the catabolism and the anabolism. So making the energy and the utilization of the energy, both are happening here. Then all we know that the healthy food it is having, these are also chemicals. Different type of the vitamins are there. In 4th and 5th class we studied, vitamins are very important for the body, which are available only in the fruits and vegetables. But here we can see, these are the minerals, these are also important for the body. Then we can see all this. What are they doing? Any chemical reaction you see, in that catalyst is there. In that catalyst you can see, you do remember that. So Grignard reaction, we can see the magnesium chloride is there in that. Like that, many are there. So these are all playing a wonderful role. If you don't take all this, the body will not, the metabolism will so disturb. So for that, whatever we are eating, every day the sodium chloride we are eating, chemical we are eating. So these are important for us. If you ignore any fruit and vegetable, we will not get the minerals and vitamins. Yes, we need the proteins, we need the proteins, we need the proteins. Everyone will speak about the proteins. What about the carbohydrates and lipids? Yeah, if you take the carbohydrates, you are becoming obese. If you take the lipids, you are becoming obese. How much you are taking? Your body says, it gives a signal to you are eating more, stop it. So along with that, then we need all these things. Here, so if that is not there, what is happening? Our immunity is going down. There here, communicable diseases are readily there, which can affect you. Non-communicable diseases, which can affect your brain, kidney, heart and everything. So that diet is very much required for us. So to see how the global cause for the death is, that injuries are 9% and communicable diseases are 31%, we can say non-communicable is 60%. So nicely we are saying it is called lifestyle diseases. Lifestyle is changed, I changed. I don't have the time to eat, I don't have the time to have a fruit. So then these are the things. So these are all in our hands. Because of this one, many things are happening here. You can see the respiratory diseases, diabetes, cancer, cardiovascular, these many things are there in that. So these non-communicable diseases these are all chronic. What is the chronic? When corona has come, it is acute. We know that yes, it is coming. It is infected. In second day, some change is there. Third day, some change is there. Fourth day, some change is there. Like that, no? The life cycle of the virus is for that. Then it will go down. Till that time, we are suffering because it is disturbing our immune system, our body metabolism and all. But non-communicable diseases are chronic. We don't know anything. We feel happy. I am looking okay. But we have started consuming many things. What is happening? We don't know. So what is happening? We don't know. For that, the simple thing is, again I'll tell you, eat the seasonal fruits and vegetables where it can give the more immunity. So for this is a burden where we can see the world over, NCDs are day to day it is increasing. Then these are the many risk factors are there. Because of the simple to say that modifiable behavioral risk factors are there. We can modify all the things. Inactivity. How many of you are having the habit to do the everyday exercise? Yoga. Yoga is our from this no Bharat. Are we doing really? Surya Namaskar. Are we doing? Minimum exercise. We are all learned all these things. Try to recollect our 4th and 5th class. Are we doing? Eat the fruits and vegetables we learned in 4th and 5th. We learn do everyday exercise 4th and 5th. Exposed to the sun, we will get the vitamin D 4th and 5th class. Why we ignoring? So lifestyle is changing. What it is changing? Nothing is changing. We are changing. Our mind is changing. So that when we follow all these things, these are all simply we can modify. Taking the alcohol, taking the tobacco and all the things, it is in our hands. So that these are all can help us to come out of all these things. So because of this one, BP, BMI, blood glucose, lipids, these are all increasing. These are disturbing our health. So then, age and gender and genetics. These three we cannot change. Age, no one can stop. So that when we are aging, we have to take that type of measurements. And the gender, the male-female related problems are there. We cannot avoid some of them. So we have to take it forward. We have to take along with us. Then genetics, any genetic defects are there. Type 1 diabetes is there, a genetic defect. So that we have to give the supplement for that. Insulin injection we are giving. We can overcome that problem. So like that, some of the problems by giving some supplement, we can overcome all these. That may be in the form of the enzyme, in the form of the chemical, in the form of the nutrition. So that genetic defects also we can, somehow we can control all those. So this way, these are all not avoidable, not modifiable, but modifiable, which are we are saying, the lifestyle changes are in our hands. So these can help us to do. So I am concentrating, so my work is on the obesity. So this is on obesity, is the world over, day to day is increasing. So what could be the reason for this? Why it is increasing? Is there here in this, we can say Indian obesity epidemic, so it is a double, day to day is going up. Why? We know that answers I am telling you. Lack of the physical activity and we don't have control on our food. We are eating. Are we eating proper food? What is the meaning of proper food? I gave the balanced diet. What is the balanced diet? Can we balance and measure and give? Then I will show that now NN is giving the education for all of us so that know what to eat. So then these are the balanced diet we have to take. For the simple example to say, seasonal fruits and vegetables is the one step. So then we can see all these things to overcome these problems. So where it is, because of this one, day to day is doubling all these things, this can nutritional disorders. So nutritional disorders, okay, because of the diet it is happening. Annamo Ramchandra antam. When we don't have the food, we pray for the God. When we have the food, we cut down. So this is a problem for us. So when we cut down, we will depend on the chemicals. The chemistry will come into the picture. Of course, chemistry is there everywhere, but the purified chemicals come into the picture. So do it is required? That is depend on us only. So that for this one, what to do? So being a geneticist, does it that chemicals are really changing my genetics, my genes? Yes, heavy metals are there. Heavy metals are intercalating agents. They go and sit in the DNA, then that can change. But we are not taking any heavy metals except people who are working in some. They take care all the things and cement companies and all. Earlier it was in the petrol bunks, there was a lead was there. Now the lead is removed. So then people were working precautionary measurements. So I work with the radioactive elements. So I take the measurement for it. So that no, these are the things will change. Otherwise it will not change. So any change comes in that DNA level, we can say it is a genetic one. So otherwise it is not. It is a diet one, what you are eating. So gene and diet are playing a role. It is influenced. The environment is influencing your genes. One is a genetic defect, second is the influencing. So the influence of that one, it leads to the problems because this is a genetically defected one. So there is a defect in this one. We in NAN also we are having one genetically defect one. It is a obese rat. That is genetically. We are defined it. Yes, it will follow the Mendel rules 1:2:1 ratio. It is a genetic defect one. But genetics one is, it is active. It is having its own problems, but it is active. When environment is affected one, so that is not active. So that it will have many problems. So this is our, whatever the obesity we are developing is the changing of the environment. What environment is there? The first is the food, the diet. The second is the whatever we drink, water, alcohol, whatever is there, smoking, and all the things are there where these are all changing. It is influencing your genes for the up and down regulation, where it can change all the things. So these are all, obesity is giving lot of complications for us. These many complications are there. So not only that, it is changing our metabolism. Changing the metabolism is, many problems are there in that. So then all the sensitive organs are affected. Then for this, monogenic obesity is there, here the syndromic obesity is there, polygenic obesity is. Maybe some gene effect is there or the environmental effect is there. So then we can see we are working on this rat model, how it is playing a role in this. So is it same for everyone? Question comes. We are not same in the hall. Genetically we are all different. So when drug is developed, most of the time we work on the male rats, male animals, not the female. So we are giving to the female. Question comes, male and female is there. Genetically we are all different. That's what no, the drug effect or the nutrient effect or person to person it will change. Not the same. But the degree or the frequency of effect is varies from person to person. So it is called as a randomized population, it is called as a panmictic population. In this population we can see variance. We can see that no, kernels. In this plant also we can see, then insects also we can see all the changes. Then flowers also we can see, these are all genetic changes. One is the environmental effect, one is the genetic effect. Even these flowers can change the color or size can vary when the environment is changed. So then, so that why this variation is coming? What could be the reason for this one? Aging is one, we cannot stop the change. So second is how the diet, market, many foods will come into the market, we cannot change. So other day one of my friend's son is telling, uncle you don't know, you are not aware about what is happening in the world. Tell better what is happening means. Future is total packed foods, ignore the cooking and all. So we have to eat only the packed foods. So simply in their mind it is sitting that cooking is not happening, only the packed food. So, okay, Zomato and or Swiggy, we are ordering, something is coming, we are eating. So that no, our psychology is changing. So that no, slowly transition is coming in this, variation is coming. Is it true? We have to put a question. Then industrialization, another one is, then we can see the clock is twisted. So then our protein is changing. Then our gene expression also changing. So these are all happening because of the change. So that diet and gene environment is deciding how you have to be. We are what we are eating, where we are living. These are all important for us. A little consciousness can give us a healthy life. So this is nice to see the photograph. Yes, it is changing. So everyone want to be like this, full fit and good. Everyone will dream. Are we really taking that type of measurements for it? There. Then we can see this is called diet gene interaction. This is the environment, this is the genetics. both are interacting, his phenotype is changing. So then you can see these are NCDs are day to day it is growing, maybe the situation may come like this. Here, then we can see the lifestyle and environmental. It is why it is happening, a big question for us. Why it is happening? What could be the reason for it? Lack of willpower or human instinct? Our willpower is changing or it is an instinct for us. For that no, lifestyle and environment, biology and genes, many are there. Lifestyle is changing, environment is changing, biology is there, genes are there. But we can put a question for it. Then we will see some examples for it, how it is happening. Many questions are there. What to eat? Every time we will put a question. That's what no, we are in the big, when we go to the hotel or the things, no, many things will come into our plates. So that no, what to eat is in the market, many foods are coming. Our lifestyle is changing, right? Then these are the life we want, everyone want, by sitting at home and watching the TV or mobile phone and want to enjoy the life. So these are all maybe, it is one is the physical activities, lackna is there, number one, whatever the food is there, whether you are feeling hungry or not, we are start eating. Because no, lucrative, food is good, we feel palatable, so that we are eating. So that leads to all these problems. So then we can see, if you go to any temples and all, we can see all these things. Is anyone is obese here? Then we can see these are the Ravi Varma paintings. Did you see anything? No, nowhere obesity is there. Then we can see till today also. I had been to the Andaman. There we can see Jaruvas. Then they looks like this only. No change at all. Then we can see here our kings used to have the big plate and all type of the food and all. We can see here. Then Mysore Raja is there and our Nawab king. So anyone is obese? I downloaded all this from internet. No obesity. Why? What is the change then and now? That changing is lifestyle, no? That time also they ate. They worked. They are physically active. So their physical activities kept them very healthy. Here, that is a lackna, big lackna is there. So let us see some more examples of this. All you know this? You know all the animals, no? If you ask great-grandfathers, you are asked your grandfather also, you say, yes. When he was seeing, when he was a child, maybe 100 years back also, squirrel was like that, now also like that. Tiger is like that, now also like that. Okay, garden lizard is also like that. Sparrow is also like that. No change at all. But we are changing. So what is happening for it? Nice, no? To see all these things. Every morning at 4 o'clock, then he say, Kokoroko, it will not stop doing all this. It is, clock is not changed. Life is changing, environment is changing, everything is changing, but clock is not changing. Here we can see, goat is like that, dog is having his own job. But when we are domesticated all these things, so these are all lack of the physical activity, forceful feeding is here. So for us also, example we have to learn from animals. Yes, lack of the physical activity is one. The second is the forceful feeding. Because of these two we know what is inside happening, the stress. The diet is giving the stress. There is no physical activity, the stress is there. Mobile we are watching hours together, how much stress it is giving. We are not noticing all these things. We can see people are doing the holding the mobile phone, they can do like this. Most of the fingers of this one is affected. So when the computer has come now, they are having the, this is called corpus something, I forgot the name. So they got the disease of this one. So you keep doing the mouse they are using. So now they are getting this no problem. The shots, hours together we are watching. So that no, this type of problems are there. Not only that, our concentration, our eye and the changing it all, I tell that also the melanin levels are changing. So this type of the lifestyle changes, which is stopping here, your willpower or instinct. Who stopped you to not to do the exercise? Anyone stopped you? You stopped yourself. You stopped not eating the vegetables. You stopped not eating the fruits. You started choosing the fruits and vegetables. Nature is giving the best thing, but we are not adapting it. So that now here, changing the lifestyle. Yes, we needed, definitely we needed all those. We cannot avoid it. Then market we can see many things are there. We know what is green, what is red here. These are the things, nice to eat, know all these things. If we eat all these things, we are healthy, all we know it. But we cannot survive without having this. Can we avoid all those? It is in our hands or not? This we can eat nicely. This can we avoid or not? The simple answer is simple, yes. It is not in our hands only. When it is not avoided, then we can see the changes will come. People used to do lot of physical activity, a cycle and all. Then now physical activities slowly disappeared. These are the cartoon. We can see 90s and 2017 how the changes come. Then mobile phone is here. Hours together we can see. That's what. So are you sitting too much? Yes. No. Here we can see transition is definitely there. When these are all here, the transition is there. Now you have to choose the road. Now your time, what you want, which road you want to select, you select your road. So for that what you need? A big confusion. Sir, how can we change it? In market everything is there in pocket, no. How can we change? Is it possible to change? Very tough, no. If you don't eat, you have to feel shame. So that no, you decide what to eat, what not to eat. This country, this Bharat want is looking at us. Because no, all Yuvata, youth is here. So young and energetic youth. So when we are strong, healthy, this country will not spend any money on you for your health. When you are sick, then they have to spend money on that. Hospital should be there. You have to eat the medicine. Your parents' workforce also go down. They have to be at home. All will be disturbed. So be healthy for this. So choose the proper route and what to eat, what the road we have to select. To avoid the confusion, don't put any question marks. Then simple thing is habits. Your habits is there. I am eating whatever is there. What is happening? All the things, lifestyle and all. What is happening? Chemicals we are eating. No. If you take the too much chemicals, it is the kidney fail comes. All we know. But headache, paracetamol. Something is there, Soridan. Something is there, Crocin. One or the other thing we are taking. We know that kidney will fail. But we cannot avoid it. Because no, how to avoid? Simple things are there. Simple remedies are there. Then here we get into my digestive system. In the digestive system, the bacteria which is useful for us, any antibiotic you take, it will kill. Can we avoid it? If necessary only you can take, otherwise no need of all these things. So disturbing the gut microbe is disturbing your body metabolism. So that no, diet and interaction, nutrition chemical interaction. Recently we got a project that is where we are studying the in silico by using the bioinformatics, nutrients, whatever the compounds we take from the medicinal plants and the drug interactions we are seeing. How it is going along with the nutrition we are seeing. So that no, it is proper to take or not. So this type of the things now by using the artificial intelligence, by using the different softwares we are working on that. So just we initiated that work. So for that no, we want to see how it is affecting all our bacteria. Then again I will come to the obesity. Transition is here. Malnutrition. Earlier the malnutrition. If you don't eat, if there is no food, malnutrition is there. But you are sitting in Hyderabad, having the proper food in front of us, we are not eating now, that is also a malnutrition. If you are not taking proper nutrition now, that is also called a malnutrition. Then you can say imbalancing. When the imbalance is there, then problem comes. So that now, here we can say the chemical, it is called nucleic acid, my DNA will change. My DNA will change, then your protein will change. How many of you are following all these things? Then visit our NIN. In NIN we can see my plate for the day. Just visit what to eat. For the day what we have to eat. Simply we mentioned it all these things. For healthy life, for healthy food. So that then if that is all the things are there, what research I can do? Now the introduction is over. I come to my research. Wow. Silence. After lunch, no? Bed. when the things are mismatch is there, I will enter into the scene. Yes, what is happening? Why people are developing the obesity? This is my question for it. Yes, genetics is there, environment is there. Epigenetics means environment is there. They are going, yes, I am genetically like this. My genes are like this. Everyone will say simple thing. So that no, when the genes are like that, you want to ignore it? No, it is the action of both. I want to say here, interplay of this one. You are looking like this. I am like this. Rajendra is like this. If you see, ask Yadanna known to me for the past 30 years. Rajendra nu atlane unnu antannu. Nenu itlane unna. Yes, only the grey hair is there. Otherwise, no. The thing is that, and thanks to my mother, ma amma cheppindi, bidda, seasonal fruits and vegetables you have to eat. ఏ కాలంలో వచ్చే పండ్లు కూరగాయలు తినమని చెప్పింది. ఖచ్చితంగా don't spend one, you have to buy a book. That is a condition for us. Every month we have to buy a book and they eat that. When the season is there, in market we can see orange is coming into the market, 5 rupees each one. When the guava will come, jamka is 5 rupees each. When we put the money, hundreds and hundreds in our mobile. So then I want to say, take in a lighter way, friend కి ఫోన్ చేసి అడుగుతాం, ఏమరా తిన్నావురా భయన్, నువ్వు అడగకుండా వాడు తినడా అయింది. So how much we are wasting? Please try to spend money on the, we are lucky we are in Hyderabad, we are blessed enough so that eat the fruits and vegetables which are in our area, what we are cultivating. So someone asked me in Nalgonda, sir, if I eat a chinna revubandu dinna pandena ante, yes, why not? It is a fruit. Eat it. Small or big is not matter. Eating the fruit is a matter. So that no, when we are not taking, all it comes into this one. We want to be like this, we are becoming like this. What could be this one? We worked on the rat model. In that rat, genetically these 4 rats are different, not same. So this is totally different, genetically distant one. So these 2 are the close relatives. This is very close relative of this one. Okay, this is a relative, this is a bit distant one, it is not at all related to that. Genetically disturbed. So distant one. So for that one, what we saw, by giving the calories, we started giving the high calories. What we required? 2000 kilocalories to run our show, the body and all the things for the adult. So that we started giving the double the amount. So the double the amount is coming from carbohydrates, only in the form of the calories. That one is from the lipids, the proteins we did not touch, one is a mix of both. So what is happening in this? Then we started looking into this one, what exactly it is happening. All these things now, so we started giving all this for that. I removed the obese one to avoid the confusion. So I am not presenting this one, it is a different story. Then I started presenting this WNN and the Fisher and SD rats. These are body weights of this. Whatever the food you are giving, the viscer is becoming obese. So that this is the genes are more prone to the development of the obesity. Their body fat is increased. Adiposity is also increased in this one. So next is followed by the SD rats. But whereas no, this rat is not having any effect on that. So that no, diet and gene are working together. Your genetics and your food, whatever you are taking. So both are working together to decide your obesity. We can see the normal control liver is there, healthy liver is like this. We can see in the high fat diet, HF mean high fat diet, when we are given, the liver is, we can see affected here. Then here, WNN. We can say, this is S mean sugar, mean carbohydrate. This is a mix of both. This is the how healthy it is, how it is affected, we can see. In the Fisher, there is no much effect. Why WNN only is affected? So that no, genetically when we are different, so our diet and gene are working together. When you feel that I am becoming obese, then change your diet. What we can change? Eating, you are eating more carbohydrate, cut down a little. Then switch on to the protein diet and all, mix up all those. So this way you can see your immunology is totally changed in this. Immunity is gone down. So when immunity will go down, then we are susceptible to the diseases. So high calorie diets, it is affected, not only that, it has leads to the glucocorticoid stress. It is giving the stress. We know only one thing. I am telling genes and diet, what about the stress? The stress is 2 types. When we eat more also, the diet inside what it has to do? Masala food, biryani and all we eat. So it is giving the stress for us. And the second is our living habits and all the things, no, we feel always, if I go late to the college, no, Yadav sir will, so he'll get angry at me. What lie I have to say? What lie I have to say? Sir, bus late, sir. Naturally we say, but how much stress we are taking? Many things are there. To give an example, if you go late to the home, mother will ask you, why late? What lie I have to say? What lie I have to? Truth ever jeopard. So this is also a stress. Economic stress is there. Different type of stresses are there. How to overcome is, the simple way is there, yoga is there, meditation is there. We can do all these things to overcome. But these are also playing a role in this, to know all these things, to development of the obesity. Not only your diet and all the physical activity, also the stress. So these are also playing a role to know. We studied how the altered glucose metabolism is changed and lipid metabolism is changed because of the stress effects. When I am giving the high calorie diets, these high calorie diets are impacted my stress levels, and it changed my glucose metabolism and lipid metabolism, and it is okay, it changed altered my mTOR pathway. So one biological pathway is totally disturbed. Disturbing of that one is resulted in the obesity. I'm giving one example. We studied many pathways and all. So this is, we can see many genes we studied. So by using the bioinformatics and all, we identified many pathways and all. We are working on those for what is the specific for the carbohydrate, what is the specific for the lipids. This is a summary of that one. So in conclusion, we can say the present study demonstrates WNN is more prone to the develop obesity associated with the comorbidities under the high calorie environment, where diet gene interaction is played the significant role in the define your phenotype. How I am looking is interaction of the both. Then this is what we can publish in the papers, and whatever the genes are there. Then when it starts, a question comes. I am looking all of you at you people. You are all PG and degree sir said no. So then, so where it starts is, it is important to know preconceptional diet is very much important. Preconception, before going to the marriage is one. After marriage, when we want to go for the children, that is the important, where it is programming. The programming of all this is taking place in the mother womb. How many of you know the Abhimanyu story? So when Abhimanyu in the Subhadra's womb, he heard about the how to enter into the Padma Vivaha, but how to come out. Only half the story is heard. So that no, it is playing a wonderful role to know. Then we studied on that, how the deficiency of the micronutrients are playing in the, so then fetal stress, it is giving what is the outcome of that one. Then maternal chromium, how it is affected your body metabolism, what is the children born to that one, chromium deficient one. So they are all becoming the fat, okay, obese at the early ages of that one. And the magnesium, what is it is affected the neurology system, your muscle system and adiposity. So that no, at that, diseases are all happening at the time of the, then when we are in the mother womb, these are all happening. So outcome comes like this. Now another question we can put, sir, when identical twins are there, what is happening? Twins are there, both are same, genetically same. So when their environment will change, no, their things will change. We can see these are all genetically same, but we can see this is the fur color is yellow, this is the black color. Here vitamin B12 deficiency. Vitamin B12 is not proper, then we can see it has become the obese and it is leads to the problems. So that now where we will get the vitamins? Again back to the first slide. Where we will get? Fruits and vegetables. Don't think that no tablets and all, these are all, once the age will come, the absorption rate is, another study is there, only 2%. When you take the vitamins and, okay, you can take the fruits and vegetables, absorption is 100%. So nature is giving, then consume more to avoid all these problems and all. So this is a Randy Jattil work. So then soon I am going to publish my work also on this area. Then now big question. Before I stop, I can put, sir, ఇంతసేపు చెప్పారు ఏం తినాలి sir? What I have to eat? Everywhere problem is there, no? కూర్చుండబెట్టి చెప్పారు తినాలా వద్దా, big things are there. I tied my hands. భయం వేస్తుంది. Already said several times, I told the answer. Eat the seasonal fruits and vegetables which are locally available to you. నీ లోకల్లో దొరికేటియే నువ్వు తినండి. So that for that, when you are tying this, yes, I feel myself. People are feeling themselves. నేను obese ఉన్నా, overweight ఉన్నా, I want to cut down my calories. started cutting the calories. Who asked you to cut? I don't know. You are cutting the calories. How much we have to cut? What to cut? Protein, lipid, whole, carbohydrate or vitamins, minerals, when, what, how much, many questions are there. So when you are cutting the calories, how it is affecting, I started looking into that, so that no, so I am not presenting the results. Here people will see their weight and they say always, yes, my weight is increased. Or they feel themselves, I am becoming obese, themselves. So cutting calories is good or bad means, I don't want to say answer. Your conscience says, don't cut. Don't cut. If says, cut, cut. We are having here the system in Bharat, we are having the Upavasa system. We are having the Nirjala Upavasam. Japan people somewhere who published in the journal that no, so Nirjala Upavasam which is giving the combating from the cancer, we are having the system already. We are not adapting that, we are not taking from our ancestors. So that now here also we can see, yes, my question has come, cutting the calories paternal. A person like us before going to the marriage, then who is feeling that he is obese, he want to cut down his calories, not the mother. Mother can pregnant times, she is having the nausea problems and lot of biochemistry will change, they will not eat properly, there is a reason for it. But the male, they are having the habit of gutka chewing, they are having a habit of the alcohol taking, they are having the habit of the smoking, they are also having a habit of cutting the calories. So if they cut down the calories, that preconceptional, what is the effect in their children? I am not studying in them, let them suffer. What is happening to their children? A person who cut down, many studies are there, female is cutting, female is not eating properly, what is happening to the children is the studies are there. But the male also a culprit. So then I started looking into this one. Soon I am going to publish my work. I am not telling you anything. So eat properly. Then whatever is there, your conscience says, when you feel I am sufficient, stop it. When you feel hungry, only eat. Otherwise, no need to eat. So then, it is giving the body will give a signal. So before I stop, then we can say yes. Here all many scientists are there in front of me. Other day I had been to the Nizamabad. We can see the color rice. We can see no. Nizamabad farmers are producing the color rice. Only one thing is we maintain the records. Whatever we do when we are crossing and all. So each plant, how much it is growing, what is the height, what is the leaf number, okay, what is the size, is the green or yellow and all the things we record. And once the spike will come, how many grains are there, so how many days it is flowering, we have the data with us, but they don't have any data, but they are crossing, they are producing the color. Really nice research they are doing, they are colorful things they are producing in Nizamabad. So that now, so then need to concentrate. They are also called scientists, I am also called scientist, you are also called scientist. Only the difference is that we are having the data. For scientists, we have to have the hypothesis, we have to have the plan, so then we have to execute it, then we have to take the data, we have to analyze the data, we have to represent, we have to document. They are not doing, we are doing. So that no, that is the difference. Here we can see our Bharat in world first, we can say, yes, Shushruta started the science here. Then we are having lot many things. All we know, no? Who is this? Mokshagundam? Sure. Then Mysore sandal soap. What do we know about him? He is an engineer. So then who is he? Jagadish Chandra Bose. Ramanujan. Amartya. Who is he? Economics. You know Abdul Kalam, Swaminathan. So then before I stop, yes, it is changed the big revolution in the green revolution is brought by the in Bharat by the study of this one. Recent times we did Mera Bharat, loudly yaar, Mera Bharat. That is, we developed it in, there is a prototype developed in the 70 days, they have given 100 days for us. Yes, we can do. So see that Vaccine War 2 movie, realistically how we worked, then you are going to see that day and night we worked for it. In bigger way it is produced by the Bharat Biotech. Then all we had the vaccine to come out of that one. And also, then we can say Chandrayaan 3. Yes, failures are there, don't worry. But outcome is successful. We are making many things so that all we are having the capacity, don't underestimate yourself. So then we can do all the things. So then some of the quotes we can see. I am the master of my failure. If I never fail, how will I ever learn? So this is, the second thing is, this is by the C.V. Raman. Then we can see, you cannot change your future, but you can change your habits, and surely your habits will change your future. Simple quote by the Abdul Kalam. Nice books he wrote, read it, and also he interacted with the 10 million young minds. Recently, there is a book is released, all his speeches are there. While speaking on the stage, he collapsed. So that no, very nice, simple things he told. So many are there people, many things said, but we are not reading the literature. Try to read, we can understand. So now I can say, yes, what Vivekananda said. All power is within you. You can do anything, and can we? Yes or no? Then this is my team. Thanks to all my fellows who are working. Half of the teams are here. Now, then we can see my funding is given by the NIN and ICMR, DBT, NIH and UK. So then we have to write our ideas, we have to competition, nothing, no one will give. We have to put the application, we have to compete, we have to present and get the money. So then it's the biggest project, is a 20 crores project, a bigger one. So then we can see here, please, I have to say, why should I say please to you? Because no, now time is changing, lifestyle is changing. Please keep walking. Whenever you find the time, keep walking. When you walk healthy every day, if you walk 20 minutes, there is a saying is there in Ayurveda. When you walk, no, one forehead will get the sweat, then that is enough for you. So it will keep you healthy. Then eat the seasonal fruits and vegetables. Then what you eat is what you are. So स्वच्छता के लिए एक कदम, keep your environment very clean. No one will tell, it is our Bharat, we have to keep our country clean. No one will come from outside and don't say other countries clean. Let us do ourselves and do. Then whenever the occasion comes, plant a plant. Take the oath. I wrote a poetry and పుట్టినాడు ఒకటి సచ్చినాడు ఒకటి బడికి పోయినాడు ఒకటి బారసాల నాడు ఒకటి నువ్వు నాటుకుంటా పోతే మేము కూడా 300 కదరా ఒక చెట్టు అడుగుతుంటది when I was on the stage హరితారం started then said ఎక్కడరా నా వారసులు ఎక్కడ the plant is asking then when the plants are disappearing we are growing we are not giving please plants పెట్టండి చెట్లు పెట్టండి మొక్కలకు నీళ్లు పోయండి then I can stop here thank you very much for giving this opportunity.
We are asking 2 questions in front of you.
Okay, no problem.
Good afternoon all. Okay, the 2 questions, get ready, my dear students. The first one is, sir had clearly explained what we are. We are nothing but what we eat. Okay? We are what we eat. Okay?
Water, yeah.
The first question is, the influence of diet and gene. The influence of diet on a gene. Influence of diet and gene. Okay, combination is a result. Result is called as phenotype. A technical word is there. Very good. Okay. Okay. Stand up. Come here.
Epigenetics. Answer is
epigenetics, but she is telling it is phenotype.
Phenotype is
Also,
outcome
also
is a phenotype.
Outcome is phenotype.
Yeah.
Okay. But actual answer is epigenetics. Okay. Genetics is one. In addition to the genetics, the influence of environment on gene is called as epigenetics. Okay. In order to maintain our body for 24 hours, how many kilocalories of energy is required?
For adult.
For adult. All are adults. Stand up, stand up. Come here. Come here.
Yeah.
Very good.
You are not listening.
Thank you, sir. Thank you, sir.
congrats. Yeah, I want to put a word. No, this is organic chemistry. Till today I can say it is on my tips. Now also I can take the class for you. All their name reactions I can tell you. Okay, thank you, Anna.
Right. Now felicitation. Right.
నిద్రపోకుండా ప్లీజ్. థ్యాంక్ యూ వెరీ మచ్. చప్పట్లు ఎందుకు కొట్టాలి? లేపేందుకు. Acupuncture. It is the acupuncture. So that it will keep us healthy. Okay, thank you very much.
Chemical composition of Western foods and what is the balanced diet, chemistry and biochemistry of the foods. And sir also explained about the vitamins and minerals and global causes of death, non-communicable diseases, and how harmful to non-communicable diseases are how harmful to the health, and remedies for that, and genetic causes of the obesity and environmental causes of the obesity, and why variations occur, and sir has concluded with a habitat to eat seasonal fruits and improve your health. With sir, your talk is very inspiring, and our students will habitate to eat these seasonal fruits, sir. Thank you very much. Introduction.
Now I introduce one more speaker, that is today's last session, that is by Dr. N. Ravikumar Reddy. I welcome sir. Ravikumar Reddy is from, I welcome Ravikumar Reddy onto the dais. Ravikumar Reddy is my classmate in MSc, as well as we did under the same guide, Satyanarayana, and we are having a colloquial relationship nearly for 30 years. Okay, he is working as a senior general manager in Suven Pharmaceuticals Limited, Hyderabad. Okay, he did his MSc Organic Chemistry in Nizam College and got CSR and everything, and completed PhD from Usmania University under the supervision of former Vice Chancellor, that is Professor S. Satyanarayana of Usmania University. That is title of the thesis. His work is done synthesis and characterization of vitamin B12 modal complexes and equilibria and kinetics of axial ligation of bromomethyl cobalaxymes. Okay, I think everybody knows that vitamin B12. What is vitamin B12? Cyanocobalamin. Okay, he did model compounds of this vitamin B12. He did his postdoctoral fellowship. After PhD, he went to Taiwan and did his post PDF in under Professor G. Wang Kem in Medicinal Chemistry at National Taiwan University. Okay, he is having 20 plus years of research experience in pharmaceutical industry. Okay, he is having 2 patents and developed by developed by CSR NCL Pune. Those patents were developed under his leadership. He authored nearly 25 publications are there and invented more than 10 patents are there. 2 patents are under the CSR NCL as well as 10 patents are there at present. Thank you, Ravi Kumar Reddy, for accepting our request, and you please continue your lecture.
Thank you, Dayanand, for your elaborated description of me. Looks like there are so many degree colleges in the city and in the Telangana state. But I think it's not possible for all the colleges to conduct this kind of seminars. For conducting this kind of seminars, there is a lot of enthusiasm in yourself, and there is a lot of hard work. And I think you people taken that that burden, and because of that only this seminar is in front of us today. And so that's why congratulations to you all. This is my first one. And the second one, how many people you are there in the chemistry now, chemistry studying? All of you are from chemistry or any other subject? Chemistry only. You know, Hyderabad is the pharmaceutical hub, not only for India, I think it is for all over world. Now in our Telangana or in our both of the states, Telangana and Andhra Pradesh, now so many pharmaceutical industries are there. And in fact, nowadays everybody is going to BTech for studying, not coming for the regular graduation and regular post-graduation in chemistry. But you people may not be thinking that there is no scope for chemistry or something like that. But it's not like that. Because of many industries, many multinational companies are coming to Hyderabad. There is a lot of scope, and I would like to request you people, you love chemistry and you try to read chemistry in your post-graduation. There are a lot of opportunities for you in chemistry. There are many different kind of chemistries also there. Now, what we know, many times what we know is only some batch type of chemistry. So we take some reactor or some flask and we will add the chemicals and we will run for some time and we will get the product. Now that is also there. Now it is also, but there are so many different kind of chemistries also nowadays is existing. You know, there is a parallel chemistry, parallel synthesis chemistry is there. So here, instead of doing for one reaction at a time, you can do up to 20 reactions in a same time, say the same timeline, you can do 20 different chemistry reactions you can do. And there is a flow chemistry. Nowadays, flow chemistry is emerging a lot. Nowadays, flow chemistry is emerging a lot. This flow chemistry, do you know what is flow chemistry? Anybody know? Anybody know the flow chemistry technology? So here actually, that flow chemistry technology is a different new kind of chemistry here. Actually, in the regular batch mode chemistry, what we'll do, we will do all the chemicals we will add in the reactor or flask, and we can run for a time and we can get the work done. But in the flow chemistry, we will do all the reactants simultaneously we are adding, and they are reacting the chemicals and they are coming out for the product. And it is a continuous process. So here, the only small amount of reactants is mixing and they are getting out as a product. So this kind of new chemistry is emerging. This is also one of the new chemistry. Not only this, there are so many other different kind of chemistries also is there. And not only organic chemistry, you can have a lot of inorganic chemistry, analytical chemistry, and regulator bodies also. You can have jobs for quality assurance department also. You may have a lot of opportunities in a regulatory department also. So that's why you need to read chemistry. So our industry is also waiting for the new talented people for chemistry jobs. That's what from our industry side I want to tell you. Just I want to motivate you people to do chemistry and all these things. So after this, just I want to go for a pure chemistry. Just now our Rajendra Rao sir told that you don't use any drugs. That means if you have any problem you use, but you have to avoid the drugs. But here actually today I am telling about one particular drug, the apremilast, and how this drug is evolved in the chemistry. It's a pure chemistry. I want to tell about this drug and how this apremilast is evolved in the this one. So here my contents for this seminar, this lecture. We have one is first apremilast API. It's an introduction. So here the drug history and what is the product background and clinical background of this apremilast API. And second is various strategies of synthetic methodology. What are the different approaches for the preparation of this API? And what are the different, here one is resolution synthetic approach, asymmetric synthetic approach, and chiral auxiliary synthetic approach. And actually our Suven has done this API and the advantages of the Suven process and our team advantages I want to inform. So apremilast is a recent active pharmaceutical ingredient. This drug is in the market, and this one presently Amgen organization is having the patent for this one. Up to 2028 they have exclusive rights, and now they are selling this drug. This brand name is called as Otezla. This drug first published in 2000, and this FDA is approved this drug in 2014, and European Union also approved in January 2015. And so that is increase the whatever the anti-inflammatory cytokine IL-10, it increase the levels of anti-inflammatory this one, and this reduces this TNF alpha and IL-23. Because of this drug, how this act is that first it binds with PDE4 inhibitor, and this reduces the psoriasis and arthritic psoriasis. This is the first cell gene in 2000 year published this literature for the synthesis of apremilast. It is a racemic apremilast. Here they taken the 3-ethoxy-4-methoxy-benzaldehyde. This one they treated with LIHMDS. So are you okay with the chemistry, or do you want? Huh? Because so silent, everybody is, there is no noise. Two things are there. One is if you get bored, then only you become silent. Otherwise, if you are really excited, then also it will be silent. Looks like the first one is the right thing. Anyhow, so this is the 3-ethoxy-4-methoxy-benzaldehyde when reacted with LIHMDS in the boron trifluoride etherate complex. Then it treated with the anion of the dimethyl sulfone, we will get the compound number 3. And the hydrolysis, we will get the racemic aminosulfone. That racemic aminosulfone, when treated with the N-acetyl phthalic anhydride, we will get the apremilast API. So this is the first published one, and based on the biological activity of this compound, they further developed. So it looks like during the 2000 time or 98, 99 at that time, this one passed the phase 1 clinical trials, and after completion of this phase 1 clinical trials, they further developed this molecule. So this is the first one. And later, after successful completion of the phase 2, phase 3 trials, this Celgene again published a chiral molecule, chiral of apremilast. Yes, apremilast is the final product. You know, in the morning also somebody told, DS Reddy shows that when one molecule, if it is a racemic compound, that is the mixture of the isomers, that also active, but when you take out the particular isomer, either S isomer or isomer, one is the very active and another is the completely less active. So here also, this S isomer is highly active for this psoriasis and R isomer is inactive. So that's why they separated the S isomer for the biological study, and this S isomer is the now particular active pharmaceutical ingredient. So this one here actually whatever the racemic aminosulfone was there, that racemic aminosulfone resolution they have done with N-acetyl leucine. By using N-acetyl leucine, they isolated the S isomer, and this S isomer when treated with the N-acetyl phthalic anhydride that produced the S-apremilast drug. So this is the synthesis. Actually, Suven has done this one in the year of 2008. Actually, Celgene given contract manufacturing for this one, and we have developed in the higher scale up to some 200 to 200 kgs. We have manufactured at that time, and we have supplied to the Celgene. So this is the racemic approach, and we with the N-acetyl L-leucine as the resolution reagent, we have made this one. And other resolution approaches also is there, published with one is some other publications in 2019. They used diparatolutyl L-tartaric acid as a resolution agent, and another is L-phenylalanine paratoluene sulfonamide. So everything here actually, the major compound is the S-aminosulfone. So here actually N-acetyl phthalic anhydride is the another reagent. So here actually the chirality, we have to maintain the chirality here, and this chiral molecule, manufacturing of this chiral molecule is the important one. And by condensation of this chiral molecule with phthalic anhydride gives this S-apremilast here. This is published in 2019 by one of the resmectmen. So here also, that both are the resoluting agent to all the three resoluting agents, they are manufactured this aminosulfone. Here, enzymatic resolution approach also have done for the apremilast. Here they used Novozyme 435 as the enzyme. By using this enzyme, by reacting with dimethyl carbonate, they have made this S-amino substituted aminosulfone. And here they used another this one methoxy acetate. They made this complex compound. So both after resolution they give S-aminosulfone. By using this S-aminosulfone, they made the S-apremilast. This is the another approach they have done for the synthesis of S-apremilast here. Here the yield is more, but one thing is by resolution approach, there is some disadvantage. That those things later I will discuss. So this is published in World Patent in 2016. It is published now. This is another enzymatic resolution aminosulfone. Then it gives the S-apremilast as our final product. So here another result, Baker's system. We got this rhodium catalyst by using this ligand, tertiary Josephus ligand. We got this aminosulfone. So this aminosulfone again with treating with N-acetyl phthalic anhydride, we get the apremilast compound. This one is published in 2015 in a US patent. This is the another asymmetric synthesis using Noyori catalyst. So Noyori catalyst is the ligand. First we will get this chiral hydroxy molecule. This chiral hydroxy molecule protected with the methane sulfonyl group. This then treated we get the potassium hydroxide DMSO, you get the this sulfone, this one reacting with this sodium tertiary potassium and phenol borane, you get this compound. Here this one hydroxy compound treated with this one, you will get the S1 compound. So this is the little different approach here. Here actually they taken the amino compound and when reducing with this triphenylphosphine, but DED and PP3 we get the S-apremilast compound. This is published in World Patent 2016. This is the one another approach. So this is the one another approach with the enhanced selective synthetic approach for synthesis of apremilast. Here they use this catalyst for making this chiral compound. So this is the chemistry. So you will get that to get this S-aminosulfone compound, and after condensation with this anhydride, you got this S-apremilast compound. So this is also published in tetrahedron asymmetry. This is the inventor Celgene only. Celgene published this compound. This is the chiral auxiliary approach. So till now we have seen that resoluting agents approach and asymmetric synthesis approach, and the last one is the chiral auxiliary approach. Here they used this amino benzol isonaphthol as the auxiliary reagent. Here they condensed this aldehyde with this auxiliary. They got this kind of cyclic compound. This one, when treated with the anion of the dimethyl sulfone, you get this kind of protected compound here. This protected compound, after debenzylation of this compound with by using hydrogen palladium carbon, you get this chiral aminosulfone. So after this, that is the regular chiral aminosulfone. When reacted with this phthalic anhydride, you get the S-apremilast. This is the approach. Okay. So here this amino benzyl isonaphthol as the chiral auxiliary, and here one naphthol ethyl amine as a chiral auxiliary. This one used as a chiral auxiliary. Here this keto compound treated with this naphthol ethyl amine to get this kind of condensed product, and after debenzylation by using hydrogen palladium carbon, you get this S-aminosulfone. Sulfinamide, generally this tertiary butyl sulfinamide is called as a L- chiral auxiliary. Bromo derivative. This bromo derivative, when treated with the sodium methane sulfonyl, you get this sulfonyl compound. Again this keto compound, we got it this one. This keto compound, when treated with this L- auxiliary, tertiary butyl sulfonyl auxiliary, you get this sodium borohydride, you will get this chiral aminosulfone. So by doing this chiral auxiliary, you get this again this one. This is the one approach. This is published in China patent in 2018. So this is another nearly 500 crores from this particular molecule. That's the advantage from last several years. So this is one. So here actually what was done, first from aldehyde, we have converted aldehyde into nitrile group. This nitrile group, when we treated with this, we have condensed with a very simple phenylethylamine. So earlier this naphthylamine also is there, some another chiral auxiliary also there, but those are very expensive. But this is the very simple chiral auxiliary, and this chiral auxiliary gives very wonderful conversion, and it gives this kind of enamine derivative, and when reduction with this one, we will get this SPA aminosulfone. So this kind of aminosulfone also, we got very wonderful yield, nearly 80% of yield, and around another diastereoisomer also only 10%, and that is also going into the mother liquors here. And this, after with the palladium carbon debenzylation reaction, we got this compound around 90% yield. So this aminosulfone, later we condensed with this N-acetyl thalic anhydride, we got the S-apremilast APA. So because of that, this is the chiral auxiliary, this one. So here, what happened? This is not working. Battery pen. ఇక్కడ మనకు వస్తుంది కదా ఓకే నా ఆల్మోస్ట్ ఓవర్ ఓకే ఓకే ఓ అయిపోయింది So here actually, just now I told you the advantages of the Suven process. Actually, up to 2028 we have the patent, and these are some advantages, because this is a very simple process with better yields and purity, and the economically viable and cheaper chiral auxiliary S-phenylethylamine we use it for this synthesis of this compound, and it has environmentally advantage also. When you see this resolution agents, by using the resoluting agents, and by nearly 50% of the unwanted isomer we have to take into the mother liquors and we have to take out. So there actually 50% completely this one, and another some yield loss around 10% or 15%. Total nearly 60% of the compound we have to throw into the waste. That is the resolution method. But by using this method, we can avoid and we can save the environment. Not only money, we can save the environment also by using that one. So, and advantage over other public process, this is already patented in several countries. Nearly more than 40 countries we have, our cell gene is patented, and even our Suven also got the names for this patent. And this chiral auxiliary method has an edge over other methods for the synthesis of apremilast, as it is simple, safe, efficacious, and economically efficient. Using chiral ligands and asymmetry hydrogenation, transfer of using rhodium catalyst is not suitable for scale-up synthesis. Here actually this chiral synthesis by using rhodium catalyst, rhodium catalysts are very expensive, one thing. And another thing is handling of that, because these catalysts are very highly oxygen sensitive and moisture sensitive, this kind of things, with their handling of that kind of catalyst in the scale-up batches also very difficult. And chemical resolution and chemical engineering, already this told, nearly 50% of mass is going into the waste. Okay. So with this, I am stopping my presentation. It is actually I want to give acknowledgement to my colleagues during for this one. Dr. Chinnapillai Rajendran was our vice president during the present this one, and these are the other my chemists: P. Ishwaraya, Amarendra Reddy, Prasad Rao, Surendra Reddy. These are my colleagues for working this compound. And Dr. Puttapathi Narendra is our present boss, and Venkateshwar Jasti was our Suven chairman. And these two people, Surendra Reddy and Prasad, also is present here in this ceremony. Thank you for all. And anyhow, it's a small presentation. I think you may not, it's having more chemistry. You may like or not, I don't know. But one here, why I came here and why I involved even as a advisor of this seminar also I am involved. We will talk about it. And Yadaya sir and Dayanand invited me and actually discussed regarding this seminar also. My important instruction is you people try to motivate and try to learn more chemistry. That is my intention. That is the main purpose to come here also. Thank you all.
So Ravi Kumar Reddy sir is working in Swen Pharmaceutical Limited. Last time from our college, more than 50 members, students, we visited that lab, that industries. Sir given lot of cooperation and also he provided lunch to all the students. This is a greatness. And also, sir, previously, at 150 years periodic table, at that time also, sir came here and given a lecture. So he's always support to us. He is just like our family member. So we give a big clap to our sir. Thank you very much.
Thank you so much, Ravi Kumar Reddy, okay, for your wonderful lecture and elaborate discussion of the synthesis of the drug. Okay, my dear students, there are two questions as usual after every session. Okay, very simple for the industrialists, but for our students. First one is, there is a catalyst that is used, that is derived from fungus, yeast fungus. What is that catalyst called as? Used for the assimilation.
No,
Students say. Students.
Who is that? Please write that. Stand up.
Baker yeast. Can I prize వచ్చిందా ఒకసారి నీకు? Correct. ఒకసారి వచ్చిందా? Okay, very good. Congratulations. Okay. Sir had discussed elaborately about the drug that is apremilast. What is the use of this drug? For you, very good. Very good. Come here. Third time, second time, కదా?
Sir, next online session tea break. Students should be here only. We are going to providing biscuits again. You will get it, and teachers and dignitaries and online start అవుతుంది. అందులో కూడా మీరు టీ దగ్గర రావచ్చు సార్ పక్కనే. ఇక్కడనే ఉండండి, ఇక్కడనే వస్తుంది టీ అంతా. భాస్కర్ టీ ఇక్కడనేనా? రైట్ ఓకే రైట్ షూర్.
I request all the dignitaries to join for the tea in Magdum Hall, in front of Magdum Hall. Okay. Tea will be supplied here only. Next session will continue.
Technical session is chaired by the Dr. Parvatalu sir.
Next technical session will be presided by Dr. Parvatalu, Department of Physics. Now I request Parvatalu to say few norms which we are expected to follow.
So good evening to all. Myself, Dr. Parvatalu Kalakonda from the City College. I'll be going to be chair for this session. Now, actually, I mean, great welcome to International Conference. So the rules are going to be the presenter should have a 5 minutes. He has to focus the main focus of the project. So begin with the introduction, then followed by results and conclusion. So he or she has to complete within 5 to 6 minutes. Then you have the 2 minutes questioning from the participants and even from the session chair. Then the number of slides varies from around 8 to 10 slides. So I request all the presenters who are going to present even physical mode as well as online mode. So we actually decided to begin with online, but a special request from the Surendra Reddy sir and I'll begin request Surendra Reddy sir to come on to the dials. And is all the students, the session that this session is going to be technical session, and you have a full flexibility to ask questions, and be patient and do not even make any noise. I can hear you sound from the last benches. Students, we don't want to hear any sound from the back benches. Title. You gonna title and name title. Slide open ఎక్కడ దీంట్లో అయితే మెన్షన్ చేయలేదు ఉందా టైటిల్ లేవు కదా So, yeah, I think you can begin your presentations. Over to you, sir.
Otherwise, you can pointer unte correct.
Recording in progress.
Slide button. Slide. Start presentation button.
This pointer, this slide.
This pointer, sir. This slide check. Okay, done.
Okay, na? Shall I start? Good evening, all. Myself, Dr. Surendra Reddy. I came from Sugan Pharmaceuticals. So I would like to say about one drug, which is how we improve the process with respect to the today. Today my topic is my topic is an improved process for the synthesis of ibrutinib drug and PCI 29732. Before going to description of this drug, just I would like to share some basic information of this drug. Ibrutinib is a proton tyrosine kinase inhibitor. It was discovered and marketed by Pharmaceutical in partnership with the Johnson Company. It was first approved by US FDA in the year of 2013. It is used for the treatment of mantle cell lymphoma and chronic lymphocytic leukemia, blood cancer. So it is marketed under the brand name of Imbruvica, and the dosage is 140 mg per day, and the patent expiry is in the year of 2026.
So my session is only how we improve the process with respect to the literature or discovery process. So before that, I would like to present the what literature says, how they introduced the drug or how they discovered the drug. Here, this is the drug. So they started with 4-phenoxy-benzoic acid derivative. Here, if you observe first, the acid is converted into acid chloride, and then acid chloride treated with melonitrile. Melonitrile, it gives hydroxyethylene functionality. And hereafter, the OH is converted to methoxy, so by using it to trimethylsilyl diazomethane. Later, this methoxy substitute derivative is converted into pyrazole derivative by reacting with hydrogen hydrate, and subsequently converted into pyrazole pyrimidine derivative by reacting with formamide. And this is the one of the key intermediate. Here, this intermediate subsequently converted into N substitute derivative by reacting with hydroxy piperidine derivative by Misnovi conditions. And further hydrolysis takes place, free NH of derivative, and finally it is reacted with acrylyl chloride. It gives a hybrid inhibitor. This is what the discovery or medicinal chemistry says the root. After seeing this, we thought that there is a wide scope to improve the yield by taking few parameters. So we were interested. If you look at this scheme, this is the key intermediate. With this intermediate, we can trigger the target molecule. So we were interested to synthesize the key intermediate by following the same process in terms of changing the parameters where it is required to enhance the yield. So to synthesize this moiety, we need to start from this hydroxy derivative. Here, the drawback of this entire scheme is the conversion of this OH to O-methoxy by using with the trimethylsilyl diazomethane. Almost 2 days required, or 48 hours required to get this kind of moiety. That too is very less, 50% yield only. So we thought that there are two approaches to enhance the yield. To introduce the same moiety, the first one is you introduce the same moiety, means OH protection by taking different alkali agents, or we introduce this new entity or new functionality. No matter it is what it is, that should be easily living by nucleophilic condensation. So in that perspective, we want to try this intermediate by changing this functionality, and moreover, we went for another literature. So how this OH was protected, in terms of alkyl or any functional group. So that this is the main OCH3 here. Here, if you look at this OHH functionality, there is no matter of how it was admins. The OCH3 is just as a living group, so it was not impact on the main skeleton of the structure. So instead of this, we can make any kind of functionality by easily movable. So in that way, we went for another literature. Alternative routes for key intermediates, what we thought, where it is enhanced the yield. So here also, there are several reagents like dimethyl sulfate, diethyl sulfate, and trimethyl orthoformate for alkylating agents to make OH protection. And another route also, the OH is converted into chloride derivative by reacting with the POCs and then reacted with hydrogen iodide to get the intermediate. In every aspect, they are getting low yield. So in that way, we were interested. Definitely, there is a scope to synthesize the particular intermediate with betterment way. So in that way, in our attempts, we decided to protect the OH by different alkylating agents or any living groups. So in our attempts, first we tried with acetic anhydride by protecting this hydroxy functionality. Here, once we have the OH-related derivative, when we trigger to the target molecule, it is unsuccessful and it gets back to the OH derivative because it is highly unstable. So this we are unsuccessful in this sequence. Now we tried another way. The OH is converted into making a sodium salt of this OH and then protected with the triplet derivative, and then finally treated with this hydrogen hydrate derivative. We got to make this molecule, but here also we are unsuccessful. Later, we just treated with dimethyl M derivative and we had the functionality. This is also one of the living group, but here we got very less yield, and further we got only 5% of yield. So again we tried for another path. Here we found that in our attempts to have better OH functionality as living group, we find out that sulfonyl chlorides are better. It works better well. So when we tried with different kind of sulfonyl chlorides in the presence of different bases, almost we have 13 to 98% conversion observed by introducing this sulfonyl group functionality by protecting the OH. This is the screening of reagents, moldations and bases, how we screen the sulfonyl chlorides to enhance the yield in the particular step. Finally, we approached that the 4-nitrobenzene sulfonyl chloride, or you can call it nosyl chloride, in the presence of different bases, we got almost greater than 98% of conversions. So finally, this is what we modified process. Here again the same process, instead of this trimethylsilyl diazomethane, we introduced nosyl chloride here. Successfully we converted and we protected this OH with this reagent, and almost overall yield is 70% we achieved. This is the way. With this same methodology, we can make one more drug. This is called PCI 297322. So with this methodology, we can make this kind of 2 drugs. So advantage of this invention is the process is simple and commercially applicable. The process does not use reagents such as TMS, trimethylsilyl diazomethane and VOCL3, which are more effluents and toxic chemicals. Consequently, there is disposal problems. The process is easy to perform and less number of steps and hence economical. Of course, the overall the yield is also enhanced, reduce the number of operations in some of the stages. And finally, acknowledgement to Dr. Veera Reddy, who is the supervisor of and Dr. Ravikumar Reddy, just now he lectured on the apremilast drug, and Dr. Veera. Thank you.
Hello. Any questions? Hello. Any questions from the audience? Any questions?
Any questions, please? Okay, thank you.
So let us go for the online now. We have 5 to 6 participants available online. So we can complete the online, then followed by physical mode.
Hello.
So let us begin online session now, technical session. I just want to remind online participants, the first participant, Archana Garipalli. Archana Garipalli. Please use your chat box whether you are Archana Garipalli, Government Degree College, Bainsa. I think sorry, I can go for online. Rafia Sultana, Pranita. Let me read the online participants first. Neeli Vasavi, Rafia Sultana, Pranita, Please put your message in the online chatting. Pranita, Dr. G. Pranita, Dr. M. Balakrishna, Dr. Deepika, Hemalata. So let me begin. First participants, Dr. Neeli Vasavi, Government Degree College, Koratla, Electronic Waste in India, a review. So please share your presentation. Please share your presentation, Vasavi, Dr. Neeli Vasavi. Available? Can I? Otherwise, I'll go for the next participant, Rafia Sultana. I think she is available. Madam, you can share your presentation. Thank you.
Am I audible?
Yes.
Yeah. Good evening, everyone. I am Dr. Rafia Sultana, Assistant Professor of Chemistry, Government Degree College for Women, Begumpet. Today I am presenting qualitative phytochemical characterization of traditional medicinal plants, medicinal herbs. This was the project work done by my student Sai Sri last year.
Madam, you have 5 minutes to go. Voice off. Sir, check it. Okay. I cannot. Madam, we cannot hear your voice. Rafia, madam. Dr. Rafia Sultana, we can't hear you. Rafia, madam, you have to unmute your voice.
So,
yeah.
What we have done is we have taken 20 different plant extracts that are available in our campus, and we tried to find out whether these phytochemicals present in these plant extracts or not. So we have done our students have done very small experiments like simple tests in our laboratory to find out the presence of these phytochemicals like steroids, tannins, starch, glycoside phenols, alkaloids, and tannins. Steroids are absolutely in all the plant extracts, but we found that tannins are mostly present in all these. And starch is present in only one plant extract. And some of these glycosides, phenols, alkaloids are present in some of the plant extracts. So what are these tannins? These are very important in reducing blood pressure. Starch, we all know it's a good source of energy. Terpenoids, they have the physiological activity of antimicrobial and antifungal. Alkaloids, they work as anesthesia. And glycosides, they help in heart failure, during heart failure. And polyphenols, they are a good source for antimicrobial acid. So these are the students who has done those tests in our lab from the plant extracts. All the plant extracts are taken from the college campus. So mostly phytochemicals, in initial years we used to use these medicinal herbs, or we used to use plant extracts and medicines, but nowadays they are synthetic. Mostly the people are depending on synthetic drugs. So what we have to suggest is we have to extract our drugs from the plants because they are a good source of medicine. So directly we cannot consume them, but we have to again go for the chemical characterization and isolate what is the required chemical from the plant extracts. Thank you, sir.
Yeah, thank you, madam. It was really wonderful to work on this kind of project. And also, it is also challenging to isolate the phytochemical from the plant extract, fruit extract, etc. But how do you control? Like, did you really isolate components? I mean, how did you isolate? I mean, as per sorry, I have a question that. I mean, we know that any plant extract have many phytochemicals. I mean, FTIR is one of the.
Hello.
Yeah, ma'am.
Sir, good evening, sir. This is Pranita, Dr. G. Pranita.
Madam, I have a
share my screen, sir.
Let us wait for question, Rafia Sultana, madam.
Yes, sir.
Yeah, I have a question, madam.
Yes, sir.
Yeah, the question is.
The question is actually we know that any green extract, any plant, there are lots of phytochemicals are available.
Yes,
How do you isolate them?
Yes, sir.
I mean, FTR can help at least to see the functional group, but isolating is very challenging. How do you do that?
Yes, it is very challenging, sir. So we have done only very simple tests in our lab, sir.
Okay. Right. So participants, online participants or offline participants, if you have any questions, you can ask. If not, we'll move into the Dr. G. Pranita, Government Degree College, Women.
Yes, sir. I'm sharing my screen, sir.
I think it's not.
Good evening, everyone. This is Dr. G. Pranita, Assistant Professor of Chemistry. Today, I'm going to present identification of adulterants from different brands of milk sample, as well as estimation of casein from. Nearly we have taken 8 brands of samples. Actually, this project was done by our students. As we all know, every day, everyone is using the milk, because the milk is containing so many vitamins, minerals, as well as proteins, like which contain vitamin A, magnesium, as well as calcium, as well as casein protein, which are helpful to the human being for the bone, as well as tooth development, as well as overall development. That is the reason everybody is using, from the small babies to older one, everybody is using the milk. But nowadays, there are plenty of brands of milks are available in the market to reach the public demand. Whether all the brands of samples, whether they are good or adulterate, we want to check by picking some of the famous brands. So we want to test some of the commonly added adulterants, like starch, detergent, vanaspati, sugar, water, urea, boric acid, formalin. Generally, these are the common adulterants in the milk we generally use. We want to check these adulterants whether they are added or not. So first one is the starch. We have taken 5 ml of the milk. To that, then by adding the iodine, we can come to know if the milk sample turns to the blue color, then it is adulterant. The next one is detergent. By taking the just 5 to 10 ml of milk, then by shaking continuously, if lather persists, then bubbles also persist till the 5 seconds, then it confirms the addition of the detergent. Next is vanaspati. By taking the 3 ml of milk, to that added HCl, then added the sugar, Then if the solution is by shaking, after that if the solution is turns to the red color, then the milk is adulterated with the vanaspati. Next one is formalin. By taking the milk and adding the concentrated H2SO4 to the walls of the test tube, if the blue-violet ring is formed, if the width of the ring, then that is the presence of formalin. Next is sugar test. To the milk, then added 2 ml of HCl, then added hydroformylation, then the red color is found. That indicates that the milk is adulterated with the sugar. Next, as well as water, if we take the 1 to 2 drops of the milk, then by keeping in the any slope, any surface of the platform, then if the water, if the milk is move very quickly, then that is adulterated with the water. Next is urea. By taking the 2 to 3 ml of milk, then adding the half teaspoon of the soybean powder, then if the litmus paper dip into that milk, then if it turns to the blue color, then that milk is adulterated with the urea. Next is boric acid. To that milk, added the 10 drops of HCl, then dip the yellow white strip, then if that yellow strip is turned to the red color, then that is the milk is confirmed the adulterated with the boric acid. So these are the generally we because these are all the adulterants generally they are the colorless as well as no taste. Generally they used to add as middle adulterants because the traders wants to get more benefit. So these are the tests we have done, as well as we also done the estimation of casein. So for all 8 branded of samples, we have taken the 100 ml of the milk sample, then heated till it get volume off. Then we added 5 ml of the slowly 10% acetic acid drop by drop. Then we get the precipitate. Then we done the filtration. Then that filtrate is washed with the 1:1 ratio of diethyl ether as well as ethanol. Then let it dry. The next day we have checked the weight of the casein by using this formula. And so these are the results of we have taken Amul, Heritage, Good Life, Cow Milk, Buffalo Milk, Country Delight as well as Arokya, Dodla. These are the 8 different samples we have taken and these are the tests we have done. So what we found is, in that case, no sample is tested positive for starch as well as vanaspati and boric acid, and 25% of the samples were positive for the detergents, and sugar was found in 50% of the sample and water in 37% of the sample. Urea and formalin, generally they are very harmful to the health, but they are also detected 12.5% of the tested samples. As well as we also estimated the casein. These are all the different brands of samples, but highest casein is we found in cow milk, and next is we found in the buffalo's milk. And and also we have done the pH test for all these samples, and what I conclude that, so every consumer, they have to aware how to check the adulterants, because what we have done, all these tests are very simple tests by adding the only HCl, H2SO4 once in a while. By checking the what they are using that milk, by checking that, they come to know whether the milk is good or adulterated. As well as instead of using any packed milk, because the cow milk as well as buffalo milk, they are not at all adulterated, as well as we also found the highest casein in that milk. So we suggest to use the buffalo as well as cow milk. Thank you for giving this opportunity. Thank you.
Hello?
Hello.
Can you hear all of you? So we don't have right now online mode session presentations. We are moving into the physical mode presentations. We'll have it tomorrow. Okay, we'll intimate in the WhatsApp group. Thank you. Right. Video first. Yes.
Hello, excuse me, sir.
Yes.
Hello?
Yeah. Tell me.
Balakrishna, sir. I have online information. Hello?
So present tomorrow, sir. Today.
Sir, shall I
Sir, Balakrishna, sir.
As per schedule, my explanation is, sir. Yes, sir.
Okay, నిమిషాలు చేస్తారు
Hello?
ప్లీజ్ టుమారో అన్నా బాలకృష్ణ
Sir, I am audible.
మేడం బాలకృష్ణ
sir,
సార్ టుమారో we are going to organizing
Sir, this is it.
today is
Hello?
tomorrow you are okay.
Hello?
క్లోజ్ చేస్తున్నారు follow.
It is coming, sir. Good evening, all. Respected Principal, sir. Vice Principal, sir. To the organizing committee and respected chair of this session, a very good evening, sir. This is Anil Kumar Bojja, Assistant Professor of Chemistry from Government Degree College, Badangpet. As of your instruction, sir, I have made my presentation for exactly 5 minutes and I have followed your guidelines, sir. Thank you. So here I am going to explain my research work that is about synthesis of novel chromin ring pendant 1,2,3 triazole derivatives. Yeah, here it is my research work orally. So here alpha hydroxy naphthaldehyde is subjected to the propargylation with the help of propargyl bromide, and propargylated product is given for the click reaction by aryl azide. This is very well-known reaction actually. This is the reaction known as click reaction, which got the Nobel Prize in the year of 2022 in a chemistry subject. The product of click reaction is subjected to the double condensation reaction with dimidone and melanonitrile to get final products. The final products are prepared in 3 steps. The first step is propargylation, which is done in a DMF solvent, dimethyl formamide, with the help of potassium carbonate. Potassium carbonate acts as a base which will helpful in the removing H+ ion from the phenolic hydroxy group, and phenolate ion will act as a active nucleophile and it will attacks at the methylenic carbon of propargyl bromide, and we will get the substituted product, propargylated product.
This is the terminal alkyne. This terminal alkyne is given for the 1,3 cycloaddition reaction between terminal alkyne and terminal azide. Generally, azides are unstable, whereas aryl azides are somewhat stable, so that the reaction conditions are maintained to the azides be in contact with the counter reactant parts. So here the propurgulated product is given for the click reaction. Copper sulfate is the catalyst here. So this is the well-known reaction, click reaction. It has great advantages actually. The reaction can perform in aqueous medium also. No need to maintain the inert conditions actually. For some reactions to prepare the heterocyclic compounds, we have to maintain inert conditions. Nitrogen atmosphere conditions, they have to be maintained, but here the advantage of this click reaction is no need to maintain the inert conditions. So here we will get the 1,4-disubstituted 1, 2, 3 triazole derivatives. Of course, it is also the novel compound. To make the work more intensified, I have given the final product obtained in the second step for double condensation reaction. So here the click product is given for the double condensation with dimidone. The compound 6 is dimidone, 5, 5 dimethyl cyclohexane 1, 3 dione is the IUPAC name, and with melanonitrile. So here this is DMAP. That means dimethyl 4-dimethyl amino pyridine. Pyridine is the base actually. Of course, it is aromatic compound due to the lone pair available on nitrogen atom. So it can donate the lone pair electron to the lone pair electron acceptors, and it can also act as the base. So dimethyl amino group at 4th position will enhance the basicity of that pyridine. Therefore, the 4-dimethyl amino pyridine was taken as the base in ethyl alcohol solvent. The reaction is carried out at 70 degrees Celsius temperature. The reaction is stirred for 4 to 5 hours on magnetic stirrer to get the final product. So this is about the synthesized compounds were established by the spectral data. So here the final compound infrared spectra I am going to explain. The peak around 1362 centimeter inverse in infrared spectroscopy suggests the presence of NH group. So the N bond H stretching frequency will appear at 3062 centimeter inverse, and similarly here the peak around 2125 centimeter inverse, 2125 centimeter inverse peak suggests the presence of nitrile group. And here also we have ketone functionality. That ketone functional group appears, functional group absorption band appears at 1666 centimeter inverse. This is the spectral data of infrared spectroscopy, and I have given the prepared compounds for proton NMR spectral data, even for 13C NMR spectral data also. Right here the spectra, proton NMR spectra of the first compound unsubstituted product. So here I have on this phenyl group I will be having different substituents. It is the firstmost compound that I have prepared without any substituent on this phenyl ring. So this is the proton NMR spectra regarding to that molecule. Yes, in proton NMR spectra of that molecule, the triazole proton, actually this 1,2,3 triazole is having only one proton at 5th carbon, because the 4th carbon is already having a substitution with naphthalene, and naphthalene which is again having 4H chromine moiety. So this 5C carbon proton appears at the delta value 8.49, and this methylenic protons appears at 5.36 delta value. This is the chiral carbon. Actually it is the aldehyde carbon. This aldehyde carbon was undergone to the double condensation reaction with dimidone and melanonitrile. So here it is the chiral carbon. it is having one proton, so that proton appears at 4.50 delta value, chemical shift value. NH protons appears at 6.89 delta value, and these two methylene groups appears in the range of 2.20 to 2.45. These methyl protons are also established with the chemical shift value of 0.97 and 1.02. Yes, sir, thank you. This is about C13 nanomass spectroscopy, and these are the chemical shift values. And these all are the compounds that I have prepared. Thank you very much.
Good evening. So respected Principal Sir and the conveners, Dr. Eluri Yadav Garu and V.C. Shukla Garu, Dayanand Garu, Mariyu Chairperson Dr. Parvatal Garu, and my dear colleagues and students. So the presentation today for this international seminar is on one part multi-component tandem synthesis, a review. So I am Dr. V. Ravindra Reddy, Associate Professor of Chemistry, working at Government Degree College, Hyderabad, Hyderabad. The next two. Point right on the point on the point on the డౌన్లోడ్ ఉందట ఇక్కడ ఉందండి ఇది వస్తుంది దీని వస్తుందా వస్తుంది ఓకే 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 వస్తుంది దీంతో వస్తుందండి దీంతో వస్తుంది చెప్పండి ఇదా ఇదేనా రైటా రావట్లేదు సో ఇంట్రడక్షన్ tandem hydroformylation hydrogenation, tandem hydroformylation with additional CC bond formation, conclusion, references. This is about this. Next. Introduction of this synthetic chemistry continually demands the advancement of more efficient and environmentally friendly techniques, enhanced reaction process in terms of high mass efficiency, minimal effort in isolating intermediate, lower energy consumption, and decreased waste generation. Generally related to green chemistry, where atomicity should be more and should have more mass efficiency, that is atomic economy should be there, and effort in isolating intermediate is not there. Directly we can convert the more reactants into products. And the adoption of tandem reaction involves a sequence of steps where each subsequent step relies heavily on the specific functional groups moieties created in the preceding step. So that is directly, it can involve 2-3 reactants can be directly converted into this. Tandem reactions are taking place, and multi-component reactions are chemical process in which minimum 3 molecules participate. So is one part taking in one vessel, we can convert the reactants into products without formation of intermediate. So this is a very good reaction mechanism. Reactions are involving in this one part multi-component system. That's why I chosen this review. Leading to the formation, and one part synthesis can be excluded in 3 main approaches. We can approach in one part synthesis, that is one part means one vessel. In one vessel and one we can carry out these reactions. Cascade or domino reactions, multi-component reactions, and one part stepwise synthesis. These 3 methods, one part reactions differ in their operational procedure. Next. So coupled catalytic processes include tandem, domino, these are already told you. The inertness of olefinic groups enable them to traverse through multiple reaction steps under diverse reaction conditions and hydroformylation conditions. I have done these hydroformylation reactions with wide array of functional groups and reagents present initially in the reaction mixture enhances the applicability of the process. And versatility chemistry of the resultant aldehyde group, because aldehydes and olefins are involving in these reactions. Next. So this tandem hydroformylation, that is hydrogenation. In this alcohols find expensive application in industry as solvents and as precursor for detergents and plasticizers. And however, the predominant industrial method for producing N-alcohols involves multi-step process starting the hydroformylation terminal alkenes, followed by purification of the resulting N-aldehydes and subsequent hydrogenation to yield alcohols. So that is tandem hydroformylation reaction. You can see this alkenes, that is alkenes in reaction with benzaldehyde. Alkene, substituted alkenes with benzaldehyde, hydrogenation, that is carbon monoxide, hydrogen at normal pressure and temperature 65 degrees Celsius. 16 hours reaction, we yield the product, and that is substituted alkane is there, and this reaction is carried out called as tandem hydroformylation reaction, and this was investigated the hydroformylation across aldol reaction of olefins. So that is aldol reaction generally we done with olefins. And next is asymmetric organocatalyzer transformations. So this method enables direct and enantioselective coupling between the two distinct aldehydes using chiral primary or secondary amines as catalyst. So there is other. And next is hydroformylation enantioselective cross aldol reaction. This is a stereoselective stereo mechanism involving in this, taking this L-proline in triphosphine phosphorus with ketone and cycloalkene, forming the substituted alcohols. And this again one more is managed reaction is also done in this tandem system, and this sequential process comprises hydroformylation of one alkane facilitated by triphenyl phosphine modified rhodium catalysts. And conclusion, in the past decade, a multitude of new reaction sequences has emerged under hydroformylation conditions, and this one of the most significant advancement in this field is the utilization of chiral enantiomerically pure organocatalysts in conjunction with the formation of aldehydes through hydroformylation reactions, and this reaction sequences provide a rapid route to enantio-rich fine chemicals starting from readily available and inexpensive olefins. So thank you. So generally, already it is done by the scientists. It is a review. Yeah, thank you.
Good evening. Respected Principal Sir Balvaskar Sir, Vice Principal Sir Shukla Sir, and convener of this 2-day seminar. Convener of this 2 days seminar, Dr. Eluri Yadaya Sir, and co-convener Dayanand Sir and other Vasanta Ma'am, Anil Sir. Today I am here to present myself, Srilata, from Government Degree College, Shadnagar. Today I am here to present trimethyl iodide is a versatile reagent for the synthesis of 4-iodo-piperidine via aza-Prince cyclization. Aza-Prince cyclization means these are the contents of my PPT, that is introduction and preparation of starting material and optimization of reaction condition and possible mechanism and acknowledgement. This is about the introduction of this aza-Prince cyclization. For the synthesis of piperidine, I have taken homoallelic amine as a starting material, and by reacting with the aldehydes by means of aza-Prince cyclization, by using trimethyl iodide as under mild condition, treatment of aldehyde with homoallelic amine in presence of trimethyl iodide at an ambient temperature, and it gives 4-iodo 2-aryl piperidine derivatives were obtained in a good yield. This is my reaction, that is homoallelic tosylamine. Homoallelic means, allelic means double bond adjacent carbon is called as allelic, and the adjacent carbon is called as homoallelic tosylamine. That can be, we can prepare by using simple homoallelic amine and reacting with tosyl chloride, we can get that starting material, that is homoallelic tosylamine. On reaction with aldehydes and trimethyl iodide in DCM solvent, we get diastereomers with one is 93% yield, another one is 7%. This is the way we can prepare our starting material, that is homoallelic amine with paratoluene sulfonyl chloride in DCM solvent. By using triethylamine as a base, we can get the starting material, that is homoallelic tosylamine. and also I prepared that trimethyl iodide by using trimethyl chloride and tertiary butylamine and DCM solvent. And this is the final product, that is homoallylic tosylamine with aldehyde and tri-tertiary butylamine in DCM. I got this product, that is 4-iodo 2-aryl piperidine derivatives were obtained in a good yield with trans selectivity. This is the mechanism. Aldehydes with trimethyl iodide. I got an intermediate, that is iminium ion intermediate. Through the cyclization, I got the trans selectivity, and that can be identified by NOE, nuclear overhauser effect. And these are the various aldehydes I have taken. Around 12 aldehydes I have taken, that is aldehydes and cyclohexanol and various tolaldehyde and paranitrobenzaldehyde, cinemaldehyde. These are the various aldehydes I have taken, and these are the the product is having high selectivity. In conclusion, in summary, we have developed an efficient synthesis of 2,4-iodo and 2,4,6 tri-substituted piperidine by means of azapine cyclization using iodo trimethyl silane as a novel catalytic system. The method was mild and selective, convenient, and the reaction conditions were amenable to scale up. All these products are characterized by proton NMR, C13 and higher mass spectrometry. And even we can identify how close are the protons, we can identify by nuclear overhauser effect. Thank you. My sincere thanks to Dr. B.V. Subbareddy sir for giving me opportunity to do my research work in ISCT. Thank you. And also the coordinator sir, Yade sir, given me this opportunity to present my paper in front of all the eminent persons. Thank you, sir.
Good evening to one and all in this seminar hall. Myself is Dr. Anjumara, Assistant Professor of Chemistry. Here my paper presentation is green chemistry. and the synthesis, calculation, and biological activity of 1,8-naphthalidine. So this is as a green chemistry, which has to be as very environmentally polluted. But when you have to be synthesize some compounds, which is has to be as very not that much of the polluted to the environment, so that it will be has to be prepared from this. So there is some variety of the compound which we have to be prepared. That is a phenylethylene diamine, in which is as to be to give as a benzene imidazole. So there is recently one compound which we have to be prepared here from this, that is that from taking as a 3RL to iso 1,8-naphthalidine and pyridine anhydride, we will have to be getting of this 1,8-naphthalidine derivative of this tetrahydrofuran, and there is some pyridine derivative which is has to be here. Then there is a present work which is we have to be did here, that is a 1,8-naphthalidine, which is taking as recently, that is a pharmaceutical, and it is very active, and it's antibacterial, anti-inflammatory, and anti-pyretic, which is has to be as to be chlorinated in that compound. So there is a one more compound which is has to be derivative, which has to be taken in a 1,8-naphthalidine, that is aryl ethylene and para-pentafluoroethylene 1,8-naphthalidine and carbohydrazide. This compound will be has to be here. So there is no synthesis which we have to be given here in this compound, but there is an some have an IR spectra which is has to be there here, this carbovizole, and that there are so many derivatives which we have to be prepared from this 1,8-naphthalidine derivative. In that there is a IR spectra which is has to be there. Next slide. And then there is a H1NMR spectra. In that H1NMR spectra will also have some 9 compounds which is there. In that all the compounds which is 9 we have, there is a one biological activity. Last slide. So there is it is most important in this green chemistry. There is an antibacterial activity, that is Vincent and Vincent. Here there is a compound which we have to be taken at 250 ml and 500 mg, and that will be as liberated into that compound, that is at 37 degrees centigrade, and that will be as very activated, and all the compound which we have to be do as the antibacterial activity. In that is a one chloro compound which is has to be there here. That is a more important, and it will be give as a more antibacterial activity. So this is as a conclusion of this green synthesis, 1,8-naphthalidine derivative of this, whatever the product which we have to be prepared in that. So thank you for giving this opportunity to the government city college and department of the chemistry. Thank you to one and all, convener and all the department of the faculty members. And also thank you for the CCC providing that the OD to the presenting of this opportunity to me. Thank you, sir. Any doubts? Any questions?
Thank you for giving me this opportunity to be a session chair. It was really a great pleasure to be a part of this program. And I mean, after this session, I think I can say there are presentations really wonderful. So yes, so all presentations definitely I can say that really wonderful. I think good practice. I think we have to always think about like innovative way to improve further. So thank you for the organizers for giving me opportunity as a session chair for this session. Thank you so much. Thank you to all the presenters who participated in this session. Thank you one and all. Thank you so much. Thank you.
I would like to thank Dr. Parvatalu for chairing the session, and he is an eminent research scholar and very much interested in research work. Now I request Dr. Eluri Adaya, convener of this 2-day seminar, to felicitate Dr. Parvatalu on this occasion. I request all the faculty members to come on to the dais. Please come on to the dais. is a great occasion to be on the dais. Please, madam, you can come on to the dais, then you can speak. Please come on. So today's program is concluded. Tomorrow by 10:30 we will gather here for the next program. Tomorrow second day session, and it will sharp by 10:30 the program will start. Thank you one and all for making this first day event successful. సాయంత్రం